So, good morning, everyone. We will start our day 2 conference, and we will start with our session 4, covering the capacity building and infrastructure development in biocuration. And I will call our first speaker. I will chair in this session with my colleague Chiama also, and with our first speaker, Dr. Cobus Horbest. Dr. Cobus Horbest is a faculty member and the director of science at the Africa Health Institute. He chairs the interim steering committee of the Africa Population Course Consortium, and he is a member of the scientific steering committee at the International Health Court Consortium and HT Africa Data and Biosamples Access Committee. Dr. Cobus is also honorary chief scientist at the South African Medical Research Council. We will not be limited to these activities of Dr. Cobus. He is also leading different activities and different scientific fields. So welcome to Dr. Cobus on the floor, and thank you. He will be covering a presentation entitled Harnessing Africa Population Course, the HPC contribution to capacity building, training, and infrastructure development. Welcome, Dr. Cobus.
Thank you, Dr. Fuza, and Blessing for your kind introduction. It's really a pleasure to present here at Biocuration. And I've learned a lot about the sort of amazing people behind the scenes of resources that we often take for granted in the scientific space. So thank you for allowing me to share some of my work and ideas with you. So I'm going to talk about the African Population Cohorts Consortium, a bit about the capabilities and capacities that we want to develop or are there within these population cohorts in Africa. I'll talk a bit about the research agenda and what the research we are promoting, what our priorities are in the cohort consortium for multi-cohort research. And then I'll focus on cohort data curation and the infrastructure and capacities we are developing within the consortium to support data use and obtaining maximum benefit for the communities in which we are based from the research that we do. Currently, we have close to 70 cohorts that are member of APCC. They're based in about 23 different countries in Africa and have just over 7 million participants in those cohorts. Many of these cohorts predates the APCC and have been following up populations for decades in many cases. So we have a rich history and legacy of data within these cohorts that becomes part of the resources that we want to manage and make available through APCC. So I want to talk a little bit about cohorts because I realize not everyone is necessarily familiar with what the population cohort is. In fact, a while ago, I was talking at the Africa Population Conference about cohorts, only to discover that what the common concept of a cohort there was a group of students that are in the same group and cohort. So that's the term cohort that they were using in the population conference. And so since then, I'm very careful to make sure that I explain. So cohorts, by the nature of the name, follow a group of entities, in our cases, individuals, over time. That's the first fundamental characteristic. The second is that there are specific eligibility criteria for those cohorts. Now, about 50 or so of the 70 cohorts in APCC use geographic eligibility. That means we are following up the complete population within a specific geographic boundary, every single person, from conception to death. People that move in and out are all dynamically followed in that cohort. It's called health and demographic surveillance systems. But there are also birth cohorts, cohorts centered around the use of a particular facility, maybe a district hospital, and then voluntary recruits, a bit like UK Biobank, all of us, where people volunteer to become part of that cohort. So from that basis, there are capabilities around maintaining the cohort. You have to decide on follow-up frequency. Our membership criteria say at least once a year, but several cohorts do that multiple times a year. Whether it's dynamic or not. And if it's dynamic, you need to have the ability to identify the events that will either signal the start of being a member of the cohort or signal the end of being the member of the cohort. And if you have a non-dynamic cohort, of course, you need to be concerned with aspects such as cohort attrition and the change in time of the eligibility construct within your cohort. Another very important and really a unique aspect of cohort is that the research participant is in a specific relationship with you, a long-term relationship. So it provides you with the opportunity to really co-create your science with the participants in the cohort. And this is something that has become more and more important for us over time, to the extent that in the governance structure of the APCC, we have a participant forum that meet at our annual general meeting and have a say and input into the governance of the consortium. There are obviously issues of consent, both individual as well as community consent, whether it's broad-based, but because we are in constant contact with cohort participants, it opens up more innovative ways of dealing with consent, such as dynamic consent and ongoing involvement in what happens to your data, your samples, as you participate in the cohort. So these cohorts have become, rather than a specific study trying to answer specific research questions, have over time become more research platforms that host a variety of studies within the cohort. And there are very few, if any, cohorts these days, at least in our membership, where there's just a single study. There are usually dozens, sometimes hundreds of studies over the life cycle of the cohort that are hosted in the cohort. These can be detailed subsamples where there's more in-depth phenotyping. Many of them host randomized clinical trials, intervention studies, policy evaluation, and then also qualitative research, social behavioral research that are done with the participants in these studies. It lends itself to rapid response And many of our cohorts pivoted during COVID, for example, to study various aspects of the pandemic, but also rapid assessment of interventions or policy changes that happens in the course of public health and other government service changes. By the nature of the fact that they last for a long time, it leads itself to look at long-term outcomes, to look at consequences of interventions that you have not necessarily foreseen when you originally developed and initiated the study. It allows for risk factor determination and correlates of protection. Then finally, a lot of secondary data analysis opportunities exist within the data that have been collected in these cohorts. And that, of course, is where data curation becomes an essential component to enable this additional value extraction from the data resources that we have. Because these are temporal data, it lends itself to quasi-experimental designs, causality studies, transportability studies, because by the nature of cohorts, they are not representative. They may be the complete population in an area, but from a national perspective, they're not statistically representative of the population. So there are then techniques to transport and to quantify the extent to which findings in the cohort can be transported to other contexts. And then itself, and there's several examples now of in silico trials in these digital twins of these populations, and then of course machine learning and big data analysis. So I'll talk more about data collection instruments, phenotype standards, and so on that we use. But imaging, functional assessment, frequency of measurement are all aspects that we have to consider. Biosample collection happens in these cohorts. Real-time data collection is becoming more and more a feature of what we do, wearables, environmental monitoring, exposome measurement in these cohorts. And then, of course, health outcomes or other outcomes by linking through administrative records to look at persons that are eligible to receive a particular service but doesn't make use of that service, to be able to look at aspects such as universal health coverage, for example. So our research programs are threefold. So first, health and well-being across the lifespan. That's really the bread and butter of these cohorts, because we follow these populations over time, disease surveillance, measures of health and well-being across the lifespan, quantify multifactorial risk factors, insights to improve outcomes. So a couple of examples of studies that are now happening and being coordinated through APCC is the African Brain Data Initiative, which is a collaboration with the Davos Alzheimer Collective to promote and collect brain health and aging data in our cohorts. And then a new study, a Gen Impact, that I will talk about a little bit more. Climate and health, if you can imagine these 70 cohorts across Africa, really provides you, across various different ecosystems, really provides you with an opportunity to understand the different impacts of climate on health, mitigating factors, and interventions against the adverse effects of weather events, for example. And then finally, something that is relatively new and I think underappreciated and undersupported currently is understanding effective universal health coverage and how the discoveries that we make, the products that are developed from our science, whether that is reaching the people where they should have impact, and what are the factors that are preventing that from happening. So about Gen Impact, we've heard in the course of these days already several speakers alluding to the genomic gap in Africa, but even within Africa, there are underrepresented populations. And this is from a couple of months ago. The landscape is changing rapidly, but the dark red countries are where there are no or very little genetic data available for the populations in those countries. And Gen Impact is aimed at those gaps to see if we can address that, making use of the infrastructure and capacity of the cohorts already in those countries. So Gen Impact, we will aim at collecting 1,000 phenotypes, 10,000 phenotypes, 10,000 genotypes, as much whole genome sequences as we can afford, together with environmental data in 5 sub-studies of 2,000 each, nested within these cohorts that I've mentioned, that also have, in addition to the core protocol, look at locally relevant applications within the Common Core protocol. We're currently in the landscaping phase, so we will release an expression of interest to all the member cohorts to tell us what they have available, how they think they can contribute to this, and ultimately we will have a call for proposals towards the beginning of next year to fund these 5 studies. This is an initiative funded by the Wellcome Trust. So now I'm going to move to the data perspective. So we know very well how to collect data in our research studies. We're really experts at that. Increasingly, fair data have become an industry, and there's lots of support and understanding of how you make data fair and what is required to do that. But there is this messy bit in the middle between once you've collected the data and you have the fair product, lots of stuff happen there, and it can become messy. And so in my own institute, this is an example of how messy it can get. We have, from our basic laboratories and so on, almost 53 million files in more than 600,000 folders, 240 terabytes of it. And I challenge anyone to really find in there what they want, except if you're the one that have created that folder and you are working with the files in a couple of those folders. Also, it is increasing rapidly. And this is what the graph is showing. But in our tabular data, in our various RedCap projects and so on, we have 600 million data items, which is number of rows times number of fields in those rows. There's longitudinal population data, more than 250,000 people, individuals, 300 million person years of observation, 10 million observational states. It takes us a long time to put together an integrated, multidisciplinary data set that can address a specific question. So what can we do to get rid of that messy bit in the middle, or at least alleviate the mess? So I think we need to have mechanisms built in how we structure, document the data to manage complexity from the start. We need to have built-in data governance and curation, not something that is bolted on after the fact. And when we do this, we need to keep in mind the 5 saves of safe people, making use of the data, ethically acceptable projects, safe settings, safe data, and safe outputs. So what we are developing and proposing to deal with the mess is the APCC trusted research environment in a box. I'll talk a little bit more about it. So we manage complexity through a data store, which is a data lakehouse on top of a data lake that manages digital assets, whether those are files, like a BAM file or a VCF file or an image or any of the myriad of different data formats that you are produced in the course of research, or it can be tabular data sets. Both of these digital assets are managed as first-degree citizens within the lakehouse. The organizing concept are studies, access to studies and the assets within those studies. We do authentication through OrgID. We explicitly store the data use ontology, the allowable use of the data at study level, or if we need finer grain data use instruction, can happen at digital asset level as well. And then also who has custody over that data, who decides who can access the data for a particular usage. In the case of tabular data, we have variables and vocabularies. The variables are linked to ontological concepts and controlled vocabularies so that we can assign meaning to those variables and have mapping of variables across different studies to facilitate access. But also a very important concept is studies are linked to entities, which are definitions of the objects that are measured or of which data is collected by the studies. And then study entity instances are linked to digital assets so that you can say, Joe Blog, I have information in these assets from these studies for Joe Blog. Entities can be households, locations, visits, etc. In your interaction with a data store, data can only come in through transformations to ingest, can only be transformed from one input assets to another output assets through these transformations. It can also be exported through transformation. Transformation keeps linked to the actual code through a GitHub commit that is responsible for the particular transformation or ingestion. So in summary then, there's a data store. It has APIs for R, Julia, Python, and other languages. And you can access it by linking to it as you would link to open a connection to it, as you would open a connection to a database from your local network. Or if you're an external user, you can access it through an analytical sandbox, which is Jupyter Hub. All of this is done through OrgID authentication, and your notebook have automatic linked then connection to the data store. And then for wider use of the data, it is exported to a data repository with a digital object identifier. Once a digital object identifier is assigned, it's recorded in the data store, and that asset becomes immutable subsequently. So thank you. I would like to acknowledge the APCC co-applicants, my Gen Impact co-applicants, and the developers of the TRIRE in the box. Thank you.
So thanks, Dr. Kobis, for this clear and insightful talk. And we will keep the question for the QA session. Thank you.
I would like to introduce the next speaker, Professor Masanori Areta from the National Institute of Genetics, Japan. She will talk on from local to global. He will talk on from local to global. Apologies. Benefits and risk of using cloud resources. Thank you.
Thank you for the introduction. I will talk about our outreach activities at my institute. Well, I'm coming from Japan National Institute of Genetics, and our institute is, of course, we do research, but our mission is to support all researchers, not only in Japan, but also in the world. So we provide supercomputing system, we provide public repositories, and nowadays we focus on human genetics too. And in addition, we also provide actual bioresources like microbes to rice and mouse as laboratory animals. And here in my institute, I have been involved in the outreach activities with neighboring Asian countries. And up until 2020, we have been focusing on data submitters inside Japan. And so we visited many institutes inside Japan to teach them about how to submit their data to INSTC or sequence databases. But as you know, we had a COVID-19 pandemic, and since then we changed our focus to a more international view, and we switched to online courses with partner country in Asia. 21 was Vietnam, 22 Thailand, then Malaysia, the Philippines, Cambodia, and we will do this year, but our partner is still under selection. And through these activities, what we realized is that at first we created tutorial movies like YouTube videos and told participants to watch these videos and please submit us questions. And in the actual Zoom meetings, we answered their questions and had discussions. That was our initial aim. So through years, we kind of expected that we can alleviate our workload step by step through such lecture materials. But in fact, as we proceed, we realized that a simple MOOC style didn't work much. Why MOOC alone does not become effective? Because as you know, giant names like Coursera or Udemy, these are giant companies providing many lecture videos. But nowadays, they are focusing on more corporate activities like short-term tutorials or short-term training courses for corporates, not for academia, not for students. Why they shift? Because academia is usually more long-term relationship is necessary. And so for companies, revenue cycle is too long. And also, to cope with academia, you need to keep up with the recent activities or up-to-date materials. As you know, AI is developing so fast these days. So every time we need to change our contents or materials, especially when we focus on bioinformatics. So cloud tutorials are quite useful, but considering the sustainability, or if you work on up-to-date contents, then simple MOOC style is not enough. So how do we respond to regional or country-by-country requests? Well, with each country, we first did a survey of what they want to learn, what they want to know about. And more we do this, more our burden will be larger because we realize that each country has its own problem. And biology is really deeply related with regional organisms, regional biospecimen and also animals and plants. So we need to learn about those local plants, animals, and also human population or diversity. So regional difference is quite fundamental. It sounds like common sense, but it took time for us to realize this. And in several Asian contexts, dominant MOOC style didn't work. For example, in Japan, Coursera is not so popular, and Japanese students prefer to learn in a traditional mentor-disciple manner. And also, Japanese people do not wish to learn through videos, but rather they prefer more face-to-face opportunities. So country by country, the atmosphere is so different. So to maximize regional activities, these are my recommendations. Of course, you need to redesign every time, not just translation, and regional specific contents are necessary. For example, we had to work on, for example, the genome analysis of national flower or regionally extincting animals and such kinds. And of course, we cannot publish every materials for free, because upon request, we are adjusting our materials. So it becomes region specific, and sometimes it may include not all public information. So, and also another important point is offline tools are so important, especially when we deal with developing countries. We tend to think that Google is available everywhere, but it's not true. And the same for other online tools. So quite often we had to provide materials beforehand, including bioinformatics tools. And they do not have full access to, cloud services, most of which are provided from the United States. So most important is the organization with multiple groups. We tend to think that participants or users have the same environment as we do, but it's not true. So of course, cloud tools are quite useful. So for prototyping and for initial questioning, initial questionnaire, we extensively use cloud-based tools. Of course, we use Jupyter Hub and also YouTube videos, but they are basically for outreach or dissemination of established contents. And especially in the initial stage, we need closer contact with organizers with each country. And we are doing this activity not only by ourselves, but also in collaboration with the ABS office in my institute. ABS stands for Access and Benefit Sharing. So this office takes care of the benefit sharing, also the legal issues between countries. So the key part is finding collaborator is in the partnering country. And of course, it's not free. So the cost and our effort is the matter. It should be resolved with time. And as it turned out that our partner countries wish to make their own repository and make their own servers, not just users. So data sovereignty is a really big issue. And what we had to do is to raise them as database managers and developers rather than database users. This is a very big issue right now. So I will wrap up. Consequence one is we need to share standards, but we need to leave stewardship locally in each country. Of course, open policy, open access policy, fair principles are very important, but what we provide is the minimum specification or minimum standard, not everything. In the yesterday, in the last session, we also heard about perfect standardization will fail, and I really agree with that view. And consequence two is we are making everything open from our website, but it's not enough. Of course, people might ask why we make everything open, but of course, in the long run, it will benefit all of us. So open access is quite important, but at the same time, regional issues, including regional issues, including personal genomes or extincting species, such sensitive information is also important. Unfortunately, many international fora, including CBD and High Seas Treaty, they are nationwidely discussed. But I want to emphasize that this nationwide discussion is too small because more regional or global thinking is important. So my conclusion is we need to really distinguish between what are to be shared and what are to remain distributed locally or in each country. So I do not have enough time to discuss about each point, but probably you will see this slide through our PDF or through the video later. So please check these points one by one. Thank you for listening.
Thank you so much, Prof, for that exciting talk. We'll leave the question and answer to the Q&A session.
Thank you so much. Now we go to our third speaker, Prof. Dr. Sébastien Baum. Welcome, Dr. Sébastien Baum, on the floor. Thank you.
So good morning, everyone. So hope everyone had coffee. I had enough, so we'll make it in time. So buckle up and we'll get started. So I'll present you today enhancing logical data, the quality, interoperability, and I want to tell you about the NFDI. So what actually is the NFDI? So it fits perfectly to the talk just we heard. It's a national research data infrastructure. We build fair repositories and offer it globally so that everyone can reach it. It's funded by the German government since 2020. We have about 90 million annually. And we have over 30 domain-specific repositories which we offer and disciplines. So from archaeology to zoology, we have all scientific data you can think of, we want to make fair. So the core goal is to develop international community-driven data standards and metadata schemas, build disciplinary data infrastructures, and have international and national collaboration. So I'm in one of these consortia, one of these 30, the NFDI for Immuno. So we are building a fair, compliant immunological data infrastructure with the international research community. We're not only covering human, but also animal data, because that's not covered very often, but now we are here to cover that. So we will only make a data repository for the data types which are not yet fair, because there are also already a lot which are already fair and meets the standard. So our target community are all the immunologists, experimental biologists, bioinformaticians, veterinary researchers, and all these kind of people. And as you can see on the map, our little consortia are spread all over Germany, whereas my research institute is in the very upper right corner. You'll see at the end one picture of the research island. So you all know the challenges. I just want to quickly repeat here. So we have heterogeneous data types. And the problem with that is we have inconsistent metadata and we have this fair gap. This results into limited reusability. And when you cannot reuse the data, then it's a waste of resources, waste of money, waste of time. And we don't want to waste anything. So what we do and what we collect, we have a metadata schema for the pharmacological data. We have a set to describe the study itself, then the subject, then the sample, when it has been taken, how it has been taken, what has been taken. Then we focus on the experiment, what experiment was done, how was the experiment performed, so that we know then the data afterwards and how we can use it and integrate it into our data sets. So the schema structure, we maintain a LinkML and a version controlled in GitHub or GitLab. And I'm also leading a little group that is focusing on the biomed consortia, and we want to harmonize and map all our metadata schemas, align all the ontologies, so that we have a one-stop shop in the end. We'll just come to one website, we'll look for your research topic, and you will find all the data sets all across the consortia, which we have and which we offer. For mapping all those, well, we heard that there are some more tools, but the ones we have identified up to that are at the bottom. But we want to integrate ontologies wherever possible, like DOID. We heard that the last several days as well. You know all the other ones, Ancestral, OB, Uberon. So that are all those ones. So we identified that if we want to have a fair set of metadata, the one thing we need to do is making the metadata submission for the user as easy as possible. So for this reason, we decided to go for an Excel template, because everyone knows Excel and has access to Excel. We'll embed all the ontologies into Excel already. So if They don't have like a free text field. When they start typing, they will be offered like all the terms from the ontology and have to decide for that, which is perfect for interoperability. And yeah, that's just quite easy. Later on, we also want to have the electronic lab notebook integration. So they basically just have to press submit and it will be uploaded automatically. And there's also an electronic lab notebook developed by the NFDI, the LARA suite, which already kind of works, but in the moment just with Zenodo and not with some other repositories. And what I'm planning to do as well is like to have a flexible input. So basically, any kind of text you have, you will just kind of submit into the LLM, which will then try to extract all the metadata and put it into our schema as we define it. So let's have a little bit deeper look in that. So the AI-assisted metadata prefilling, as already said, like all kind of inputs will be put in there. The LLM will extract all the metadata which is recognized, will map it to the ontologies, to our metadata schema, and prefill the template. And after doing that, it will send it back to the user. But because in the end, the user is responsible for the data he submits. So he has to check his data. It's just like a help. Once he has like corrected that and put all other metadata in there, he'll submit it again to our repository for quality control, and we'll have a little bit of a deeper look into that now. So currently, it's more in the concept phase. I did some tests which actually worked quite nice. The better and more expensive the LLM, that better it worked. Unfortunately, I'll try to make it a bit cheaper now. Also, we are using like write field for the Excel, for the ontology embedding into the Excel template. And the quality control system, which we'll hear now of, we already have set up and it's working quite nicely. So in the first stage of the quality control, we'll check for the syntactic validation. And that is done in LinkML because that works perfectly. We look like if all required fields are completed, the correct data types have been put in, cardinality constraints. Yeah, the ontologies will be checked again because you can misuse it if you really want it, but yeah, errors can occur, and this will help us to get rid of them. In a second stage then, because LinkML is like cannot do all the semantic validation which you want to do, we'll then convert this into schema pack. In schema pack, we have more options for the semantic validation, which we'll do there. We look for semantic logics of all the metadata, cross-entity linking, if the X to N logic is like working and if everything is kept in order. And once all this is done, then it can be actually uploaded into our repository. Well, not yet, but it will be in hopefully a year's time. So the NFDI for Immuno repository is in S3 object storage. It's cloud-based with access control. And it should support all the immunologists worldwide to get his data fair and get uploaded. So the animal data are completely free. That's no problem at all. You don't even need to log in or anything. So there's no GDPR issues, which I'm really lucky for. But we also have like human data and sensitive human data. And they, of course, have like in data access. So you need to be registered, you need to be verified. And then once you've identified the data you're interested in, then a contract will be set up with the data controllers, of course. That is all like we have standard templates for that in place and should go more or less like really streamlined. So it shouldn't be a problem. Basically, as a user, you go to the repository. Once you've registered and you've been verified, then you can contact the data provider, request an agreement, receive, sign it, and download it. Should be quite straightforward. Good. Then it's a strong coffee. I still have some time. Good coffee today morning. So summary and outlook, key takeaways I want to keep in mind is that the NFDI, so the German National Research Data Infrastructure, it's a German initiative to make like all scientific data we have fair. NFDI for Immuno wants to do that for the immunological data and for the ones which are not yet fair, there are already some. So we are working currently like on 2 data types. And we want to make it the submission of the metadata and data as easy as possible, because only then people will use our repository. So we are doing that with the AI-assisted prefilling of the templates. That will also reduce curation efforts later for the people, so for us. And we have a 2-stage validation for the quality control so that all the errors will be reflected back to the user and we have to do less. when we like, and we have like a wiki with all the error codes so that they hopefully can figure it out quickly themselves. Good. With that, we have like about 40 seconds left for a small question. Otherwise, that is the island where I'm working on. So the island of the band scientists, which is quite nice to work for. Yeah, a short question or just enjoy the view.
Thank you so much, Dr. Bohm, for your insightful talk. I need to precise that Dr. Bohm is affiliated to the National Institute of Genetics it's in Friedrich Leff Institute in Germany. Thank you for talk, and likewise, we keep you QA for the panel session. Thank you.
Our next speaker for today's session is Professor Nicola Mulder from the University of Cape Town, South Africa. I'd like to add that I've been following her work for the past 10 years of my bioinformatics computational biology career, and maybe this is like some sort of fangirl moment, and I'm so excited to introduce her. She's going to be talking on the African Bioinformatics Institute, a Pan-African initiative for bioinformatics innovation. Thank you.
Thank you, Blessing. Okay, so yeah, it's my pleasure to talk about the ABI, or the African Bioinformatics Institute. I'm going to talk a little bit about how we got there, what were the past initiatives that got us to where we are today, and then for the ABI, what is the aims, the structure, obviously the capacity building, particularly for the biocuration community, and then some upcoming projects. So the past decade or more, probably 13 years, we were working on H3A BioNet. So this is the Pan-African Bioinformatics Network from the H3 Africa project, which was a Pan-African project to build capacity for genomics, human genomics research in Africa. So in H3A BioNet, we spent a lot of time working on building the capacity for data. And that goes everywhere from the data collection mechanism. So you heard from Catherine yesterday, the data standards that we were using, data transfer protocols, when you have poor internet, data storage, security, the workflows, etc. So what we did in H3A BioNet was looked at built these instruments for SADA data collection. We used Global Alliance for Genomics and Health standards all the way along. And then we built the computing infrastructure in terms of having a local archive. It was a data coordinating center and the liaison with the EGA and the ENA to be able to submit data to those repositories. We put in modest computing infrastructure across the continent. And then we also built Nextflow workflows and containerized them. And this was through a lot of hackathons and training and capacity development in workflow development. So the training, if you think about the curator and the data side of things, the curator needs a whole workforce around them. They need the sysadmins to have the data infrastructure. They need the workflows developed by computer scientists, and then they need the analysts. So we had to train in all aspects of this. We trained, I think, I've lost track now, somewhere between 6,000 and 9,000 people, individuals across the continent over about 12 years in all these different aspects, depending on the audience. But the funding ended in 2024. All good things must have come to an end. And so we needed to think about what was next. There were ongoing needs. So we ran this annual Introduction to Bioinformatics training course, which took on sort of 1,000 or more applicants per year. Every year we receive more than 6,000 applications for this. We keep thinking, surely we've covered the continent by now. Apparently not. Our summer school, we're having a summer school for ABI. We have 30 places and we've got 800 applications. We have ongoing requests for training. We finish one course and immediately the emails start about when is the next course running. And so we have an insufficient skilled workforce that really can take data and turn it into impact. And this training needs to continue. We need to sustain and expand what we've built already in terms of the data and compute infrastructure. There's a growing demand as the continent is starting to generate more and more life science data. And bioinformatics just remains that bottleneck that we have to address. So the solution was to form this African Bioinformatics Institute, and we were fortunate about a year ago to receive funding from the Wellcome Trust and the Chan Zuckerberg Initiative to make this dream a reality. So the aims of the ABI are, severalfold: training and capacity building, so to continue that legacy, support life science research. So whereas HJ Bindit was very human genomics focused, this needs to cover all areas of life science. Promote translation and innovation. So how do we get a real output, tangible output from the data? And then this data sovereignty. How do we support locally developed tools and data repositories? And so what the impact is, high quality precision medicine, high quality pathogen surveillance, One Health, and then this building of the bioinformatics community and the skills around that. So the stakeholders are not just the bioinformaticians in the middle who we want to train, get more bioinformaticians who drive the field of bioinformatics forward, but then there's also the user base, the clients who just need to run a workflow. They need to be able to interpret their own data. And this is both in the academic and public health space, as well as industry. And then, of course, we have the other stakeholders, which are our IT infrastructure, cloud providers, software companies, the funders, and hopefully governments. And then the bottom is showing that it's across all areas of life science, no matter what the organism, and all technologies. So we looked at different models. There's obviously the EBI model, which is bricks and mortar in a central place. But because we're now dealing with 54 countries, we actually went to the Swiss Institute of Bioinformatics model, which has, even though it's one country, it has different sites around that country. So we ran a call recently. We've recently appointed 32 group leaders, 9 associate group leaders across 12 African countries. And this is an ongoing process, so people can apply at any time. And these are group leaders who are driving the field of bioinformatics forward. They're not the bioinformatics users. They are the bioinformatics researchers and service providers. What we're trying to do in parallel is work towards an intergovernmental organization like Elixir, where you have more scope for long-term sustainability. So we need to, in ABI, develop and support the data infrastructure for big data projects. So we have different kinds of services. So we always have the people having their data set that needs analysis and support for that. The African data resources, you've heard about some of them yesterday, you'll hear about some more today. We have fantastic bioinformaticians developing novel tools. And we need to make these available with an African label on them. And then we need to continue to build the data and compute infrastructure for large data-driven projects. So building on the Elixir model, where they've got these different platforms, they've also got interoperability. We'll start with the 4 platforms because we'll incorporate interoperability in the data tools, compute, and training. And I'll focus today, because we're in Biocuration, on the data side of things. So we have, I think you've heard many times, we have a scarcity of African data. Where it does exist, it's not necessarily easy to use or even able to, you're not even able to find it. So we need to get from this raw genomic data into a state where we can interpret and actually translate that data. So to do that, we need curation of large heterogeneous data sets. What are the data sets? What the technologies are coming from? What do they mean? What is the metadata associated with them? The phenotype data. We need to transform those large data sets into usable formats. So don't just give raw BAM files with a phenotype file. Have them connected. The African Genome Variation Database, which you'll hear about later today, is a curation. It's aggregate data. And then we've also, for the existing data that somebody else generated, how do we extract that and curate that from the literature and from relevant databases, where we can highlight and go into deeper granularity in terms of the African data? Because when you go to Nomad, when you go to many of these different databases from GKB, it just says African. 54 countries, huge diversity. It very much depends on where you are in Africa as to whether that data is relevant to you or not. So there's a huge requirement for data curation and stewardship. So some of the example biocuration activities, and I'm biased here, I'm focusing just on the resources that I know about, but there are many others across the continent. So the AfriGenD, I think you've heard of quite a bit about. There's the African Population Ontology, the Genome Variation Database that you'll hear more about, and others, and including the data collection toolkits. And then several years back, we worked with the Sickle Cell Consortium and community to build a sickle cell ontology, again, because the existing public resources didn't solve the particular needs that we had. In all of these, we're trying to address data sovereignty. So we asked people to submit their data to Nomad, and there was a huge hesitation to do that because it's an American resource. We created a kind of a local Nomad, and we got the response in terms of getting data. And I said African ownership that African sovereignty is still available, it's still public and interoperable. We're working with Federated Nomad. The other thing we're trying to do is increase access by translating into different languages, our websites, our ontologies, and very specifically our training materials, so that they have access for French, Arabic, Portuguese, and Swahili populations. We're also thinking about making data fair, so large-scale data management and metadata kind of finding and access. And so the Awazi catalog you heard about from Mugimi Atwini yesterday, he talked about his metadata harmonization tool, which is in REDCap. Big data sets can put their metadata into this metadata harmonization tool and into REDCap, and then it gets slipped into the Awazi catalog, which means that you can find these data sets. So there's a level of curation at the metadata level, and then we still need curation of those individual data sets for use. So in terms of developing biocuration capacity in the Institute, so the challenges are, I was a Swiss curator and then I was an Interpro curator. It very much depends on the resource you're using, and the curation tools, the data types, everything is very specific. So it's not that you can do, okay, we'll just make you a curator for anything. So you need to have specialized training. It depends on the tools and the data, and the long-term career prospects are very slim on the continent. So we can introduce some generic data curation training, like how to clean data, how to, what is data stewardship, ontologies and standards and that, and then have specific training for specific resources. We definitely need to do training on the use of AI for curators. We've had these EBI knowledge exchanges where we've been able to work with the curators at the EBI and the developers to gain knowledge about their curation experiences. And we're developing a community of practice, and we run a regular research data management course. So just to end then, you heard all about the APCC and the Gen Impact project from Quibus, so I won't go into those, but those are going to bring more data. Those are going to bring more curation needs. We also then have to extend to the plant breeding community, local biodiversity data. So our curation capacity needs are going to be increasing. And we're also exploring the potential for community federated EGA and local ENA as well. So lots to do, but we have a big team to do that, and we have the community across Africa. And I'd like to just acknowledge the core staff, Manadia and Verena and Samir, and then some of our consultants and our interim council who's been helping to govern to date. Thank you.
So thank you so much, Prof. Mulder, for this interesting presentation regarding the EBI. And now we will call for the speakers for the Q&A session. Please welcome to the floor. So any questions to the speakers, please?
Thank you, Tony. Thank you so much for the speakers. Very interesting. So I have 3 questions I will start with Prof. Kobus. So you mentioned that you will at some point you will focus on some disease of interest. What kind of disease are you planning to work on? I mean, maybe there was some kind of priorities in the continent. And the question for Professor Arita regarding what's your thought about the AI? I mean, some kind of platform for training like OpenClaw and ClawBio for training. Is that would be some kind of good solution for locals or maybe internationals? And finally, for Sebastian, I would like technically, how do you map the ontology in the Excel file? And finally, for Nikki, I have no question because I'm part of the ABI.
Okay, about the diseases. In IPCC, we're particularly interested in the intersection between the major infectious diseases in Africa of HIV, TB, and malaria with non-communicable diseases and how that impacts on aging across the lifespan.
Okay, for my part, regarding the AI use, inside DDBJ, inside Japan, our curators are using paid services because free services, for free services, we cannot use personal information or confidential information. So we always need to use paid services guaranteeing security. But we cannot tell the same for neighboring Asian countries because, first of all, their internet environment is not the same as ours, much inferior. So what we tell them is to first develop curators prior to AI use. That's the current situation.
Yeah, thank you very much for your question. So actually, for the biomed consortia, we are currently at the level to map the metadata schemas and not the ontologies yet. But there's a base service in NFDI which takes over, has developed some codes and programs to actually map ontologies and have an overall spanning ontology mapping. There are also tools online already from I don't know where, so we've got a lot. What I can tell you about metadata schema mapping is it's impossible to map 100%, absolutely. So we try to focus on the top 15 metadata, which will absolutely harmonize, and you will be able to identify all the data you're interested in.
Yes. Yes, Monica, please.
Thank you. First, I want to say, blessing you and I fangirl together over Nikki's work. That's awesome. I have a question for Sebastian. Monica Munoz from University of Colorado School of Medicine. I'm a big fan of LinkML. I work very closely with a lot of developers that have been working on this. So I'm very curious to learn from you. You were saying that Schema Pack has better capabilities for semantic mapping, and so that you use it as you export LinkML into Schema Pack to improve this. Could you maybe share a little bit of your perspective of what's lacking in the schema to help you do that mapping, or why Schema Pack ends up doing a better job?
Yeah, I'm not an expert on that aspect, but yeah, the semantic validation is in LinkML not as good as in Schema Pack. Schema Pack does have some limitations as well, but in combination, then we'll get a quality control, which we find acceptable when we will get rid of most of the issues. So yeah, we'll take the best of everything and mash it up and hope that it works out.
Brilliant. It's a good strategy. I'm curious and want to copy that. Cheers.
Yes, Alan, please go ahead.
Okay. Yeah, so thanks. A question maybe to Quibus, and then if there's time to this second speaker. And I think Quibus, I probably fall into that group of people that you encountered who are very naive about their APCC. And so probably a question that I have is, if people come to your repository and wanting to utilize those cohorts, data sets, would there be some mechanism of assistance in trying to refine the specific control group in trying to utilize that cohort? Or do you find that your users normally come there already fully appreciating how they need to fine-tune their control group in order to maximize the use of your specific data set? And then maybe a second question for my colleague from Japan is, I know you had no time to go through that last slide, but I was trying to interpret in that table what gets shared, that sort of you striking that, and I imagine yours was about the trade-offs. And so I was wondering how I should interpret your column about to be shared versus to be distributed. Thanks.
So one thing that I neglected to point out when I was talking about the TRE in a box is that this is at individual cohort level. So this is our strategy, to first provide the tools at cohort level to manage their data, to add value to their own process, and then build on top of that, using the African Bioinformatics Institute and approach, a federated infrastructure to federate data from the cohort level. So in terms of discovering the cohorts, APCC is in its first year of existence, so we don't have that capacity. The ideas that we have that we're busy developing is If you're familiar with the International Health Cohort Consortium, IHCC, they have an atlas of cohorts, is to develop a resource like that on our portal, this IPCC.Africa portal, where people can then see at that level, it's open to all, what data is available and at cohort level, and then be able to either go and look at federated resources if they're interested in multi-cohort, or go to the TRE of the specific cohort if they need more detail.
Thanks, Dr. Kobus. Vicky.
There's a remaining question for me. Well, first, I can share my slide with you later. And the distinction is the category to be shared refers to guidelines and also protocols or rules. Until now, for example, at INSDC, we have been accumulating all data to our repositories, and we consider external people as users, so to say. But through the communication with Asian countries, at least I came to realize that they are not users, but they are collaborators. So they want to have their own databases, they want to establish their own repositories. So it's exemplified by the EGA network, for example, data are distributed, and what we provide is guidelines or rules according to GDPR. The same is needed for not only human genomes, but also other genomes too. That's my distinction.
Thank you. Any other questions? Please, Vicky.
Great. Thank you so much for the interesting panel. I was just wondering with the APCC, and I'm one of those that is also coming from a place of ignorance. I'm just wondering, how are you dealing with variable consenting in your cohorts? Because, you know, some people have got broad consent, some people have got tiered. How are you dealing with that in the cohort? And also with variable laws in terms of data governance, because there are some countries that are very strict in terms of how long you can store samples for and what samples should be used for when, and some we are experiencing in the SECO in Africa consortium that some countries do not want you to store samples for general studies. You only store them for the specific study that the people consented to. They don't want broad consent. And then I guess Nikki clearly articulated sustainability, and that's becoming a very key question, especially given the funding environment that we are currently in. I would be interested to hear from the other speakers how they are dealing with sustainability of their training or their projects. Thank you.
Yeah, you highlighted a really complex area that is central to what we are trying to do. So we have a work stream led by Dorcas Kamuja from the Bioethics Network based in Kenya that is dealing with this issue to develop consent guidelines with input from our participant communities that I've mentioned, with the aim of, I don't think there will ever be sort of one consent standard. It will depend on whether it is in a particular cohort and the local requirements, multi-cohort needs. So it's a complex situation. So we are developing components. We're working with ethics councils and committees in the different countries to look at reciprocity across countries, data sharing. We have developed a data sharing framework agreement where you can lodge your framework agreement, in this case with our host institution, Science for Africa Foundation, and then you can have entries for specific instances of sharing without going through a complete agreement process in terms of the framework agreement. So we are developing mechanisms like that to ease the complexity of navigating these different statutory regimes that are in different countries.
Thank you. I have one more question related to the question of Vicky. For the period of storage of samples in labs and biobank, lacking of biobank, is it the same, or for biobank we can keep the samples more or for forever?
I think that depends on the national jurisdiction, and it is at the moment we don't have any specific guideline in that regard.
Okay, thank you. Any more questions for? Yes, Aline.
I
please.
think perhaps we have a question for Nikki so that she doesn't leave without answering a question. I just think of the ABI, as you pointed out, it is earmarking a range of fields beyond human genomics. What is your vision for how we roll out a curation working group consortium that can work alongside these various domains?
So in the ABI, we've got these communities of practice that have been set up. At the moment, they are topic-based, like rare diseases, metagenomics, pathogen surveillance, etc. And those are to bridge the gap between the bioinformaticians and the bioinformatics users. So, and anybody else, industry that needs to use them. So I think, so the aim is to create we were going to create a data stewardship working group, but probably a community of practice that can cut across those. I mean, we can't expect them to attend every single one of those, but hopefully, they will attend the ones that are within their domain. And so within this community of practice, we will, hopefully have training in biocuration. We will have their connection to these specific kind of data types, and then more of these knowledge exchanges. So at the moment we have them with EBI, which has been working very well, but we could certainly set that up with other resources depending on what that domain needs. So if there is a domain that's actually going to be a local ENA, then have a knowledge exchange with that particular resource. So I think the first thing is to just get a community together, because a lot of people are isolated at the moment across the continent. They feel like they're the only curator, don't really have anybody else to talk to. So having that community, I think, will start, and then they can break out into whatever activities they think will help that community.
Any more questions from the audience? So if you have any, don't have any, Shaima, have a question to Dr.
Thank you so much to all the speakers for all the interesting talks. I have a question, actually 3 questions for Professor Nicola. So as someone also involved in capacity building back in Nigeria, coordinating the ISCB RSG Nigeria team, I have noticed some gaps in training. Sometimes we conduct training workshops for them, and then after the training, we see that they don't take the skills and use it in the long term. So I think one of my questions would be, what do you think, or what do you believe has worked in building sustainable bioinformatics infrastructure that people don't talk about enough? And then the second would be that, do you think Africa's biggest gap is talent, infrastructure, or coordination? Which one do you think is limiting us the most right now?
So on the first question, so we encountered this quite a lot when we were doing training. People would come after the course and say, well, I still don't know what to do. So the way we got over this was that within the course, we tried to make sure that the environment that they're using, whether it was an analysis environment, was containerized, or it was kind of everything on a VM, so that they could take that home with them and immediately run it, or bring their own laptop, set everything up, so that they didn't feel like they were on their own when they got back. The other thing was to be very careful in our selections of people that were immediately going to use it. People who said, in a year's time, I'm going to have data, we didn't take them, because you forget. And so you need to be able to implement it immediately when you go back. The third thing we've been implementing for pathogen surveillance training is a trainer-trainee help desk, so that when they go back, they actually have a group of people they can ask, so that again don't feel isolated, because the lecturers move on and it's really hard to get access to them. So we build Slack groups for the community for that training course, so that they can ask their peers, and then we have this trainer-trainee help desk that they can continue to get longer-term mentorship. On the second question, we don't have a shortage of talent. We have so much talent on the continent. What was the second thing? There was coordination. What was the third? Infrastructure. Infrastructure, I think, is the biggest. So there's some coordination, but there are actually a lot of duplicate efforts that are happening across the continent that could be better coordinated. But not everything has to be coordinated. There are independent projects that should continue, but it's the infrastructure. Not everybody has stable internet. Not everybody has access to computing just to run an LLM. And trying to get onto cloud is really difficult because not everybody has a credit card to get a billing account or even money to pay for that. So I think infrastructure is the biggest rate limiting step.
Thank you so much.
So thanks to the audience and thanks to the speakers for their insightful talks. So maybe we will finish and wrap up this session and go to the coffee break. Thank you.
Maybe just a small announcement as well. I'll try to announce this multiple times today, but we've got the conference dinner tonight, and the bus will pick us up at 6:30 from the reception hall. So please, if you've got a ticket and you've signed up for the dinner, try to be on time for the bus tonight. And then also we have 2 tickets left, so anyone still interested in the dinner, please see Tanyan at the Lighthouse.
No, it's not mine. No.
We're all here. Let us know when can go. Okay, thank you. Yes, you can hear me, right? No, no. That was on purpose. So, I hope you had a nice tea break. The coffee was delicious, and also the canapes were really nice. I'm really pleased to be here today, and I'd like to thank all the panelists and everybody here on behalf of Lyndon and thank God and myself, as we have prepared this panel on career progression. And I'd like to start by introducing or reintroducing our panelists. I believe you've heard from all of them, but let's just quickly mention Professor Dr. Nicola Mulder. She's the interim director of the African Bioinformatics Institute, and she mainly prioritizes, strategizes, and builds programs on training and capacity development, and also from the University of Cape Town in South Africa. Marina Di Stefano, she is Director of Clinical Genomic Operations and PI of the Clinical Genome Resource at ClinGen. Mainly directs and trains the biocuration teams for ClinGen at the Broad Institute in the United States. Mónica Muñoz Torres is Associate Professor of Biomedical Informatics for the University of Colorado a director of Computational Health Intelligence Lab, Clean Fennel, co-lead in GA4GH. Her role in Biocuration is helping curators prepare their data to make it AI ready. Peter Welts from the Virginia Commonwealth University in the United States. Hello. Mr. Gabriel Algunas, apologies if I mispronounce your name, I'm very bad. Curriculum advisor for the life sciences and natural sciences at Metro South Educational Department, and he has influence on curriculum development from grades 8 to 12. It's Western Cape, South Africa. Barbara Strazzi, a team coordinator at Campbell, and she manages the Campbell database, including the Biocuration team at Campbell UDI in the UK. And Victoria Nunez, postdoc at the Department of Biomedical Sciences and the lead curator for this broad database from the University of Padova in Italy. I'd like to let the audience know that we'll take Q&A, the questions at the end, or questions or observations at the end. And I'll start first. We'll go through what would be a natural career progression and take it from there. So let's talk about the entry pathways towards biocuration. So I'm addressing first Gabriel, because he has the influence on the early learners. And I guess that as a personal curiosity, it says how I am aware, or at least according to surveys, the public perception of scientists, it's that we are a weird bunch. So how can we as a community encourage high schoolers and early stage learners to pursue a field such as sciences or biocuration?
Yeah, thank you very much. Good morning, everybody. From the school side, I think schools play a very critical role in intentionally integrating emerging fields like biocuration in the existing curriculum that we have at schools. Fortunately, when it comes to life sciences, now, if you're not aware, it used to be called biology, rote learning, and then we shifted about 20 years ago into life sciences, which basically includes all these different fields. So we can actually say that DBE and the WCD Western Cape Education Department, we've already put our fingers and toes into biocuration by working with different data sets and stuff. And what we also have now is that these learners, when they are being assessed, they're being assessed using the scientific method, taking different data sets, even though some of them might be a bit outdated, but at least they get the opportunity to work through it, and that also can be scaffolded when they go study sciences later on in their career. So it's important that as educators that we use case studies, data-driven investigations, use that in the classroom and link it to what the clever people here do in the room on a daily basis. I must tell you, initially it was not that easy for us to change from biology to life sciences. For many of us, we had to go for training, and currently we are still busy training the new educators going to the schools. Notably, like I said earlier, for life sciences, we have what we call the CAPS curriculum assessment policy statements, which stipulates the minimum requirements that must be covered in the classroom. Now, many times people just think when you look at the curriculum, you must cover A, B, C, and D. However, it leaves scope to be flexible. And that is where I thrive in. When it comes to the flexibility, like we have yesterday, we had the 14, 15-year-olds here yesterday. And I must tell you guys, it was a life-changing experience, Professor Mulder, for them. Already they said they will definitely pursue a career in sciences, and they will take mathematics and physical sciences and life sciences when they do grade 10, grade 11 at high school and grade 12. However, what I want to say is that there is this flexibility to change the curriculum that covers your needs at your schools. And that is basically what I am promoting. I normally say I'm a person, I like to move into the gray areas, which is not wrong when it comes to education. By doing that, I expose the learners to these things. And what I've noticed was that a lot of these learners who has gone through these things in collaboration with Professor Nicola Mulder here, Victoria Nimbawari, and also Ambrose. I don't say Ambrose here, and thank you very much, Lyndon. I've seen in the past these learners that has been exposed to these activities, they actually follow career in sciences. And that is basically where it is starting, having this 14, 15-year-olds here, encouraging them to go study sciences, and they can maybe be the next scientists who will sit here in this room in the future. However, what we've also noted was that we need skills to pursue this career. We need skills to prepare the learners to go work in biocuration, to help you guys setting up data and filling in those missing gaps. And I really think these exposures that you guys afforded us will play a big role in that. Now, in the past and even currently, we have a lot of partnerships with many HCIs, but unfortunately, in most cases, we are working in silos, where we basically prepare the learners just to finish the grade 12 exam. That is your exam that at the end of your school career, right? And we prepare them for that. And when we're done, okay, now you go to these HEIs, higher education institutes. But we were so fortunate to have made contact with Vicky and with Professor Mulder here on my right-hand side, where they actually came in 2018, Victoria, and they said, look, guys, we want to partner with you. Now, in the past, the people coming in to partner with us, it would almost like be a checkbox for them. No, we've dealt with the education department, done that, right? But in their case, they came in, they worked with the learners, we had quiz competitions, we would have the whole Metro South Education District, over 5,000 grade 12 learners coming in, and from the initial stages, we would have a competition, and the main aim of this competition was to expose them and encourage them to pursue a career in sciences. And what I've noticed, what I said earlier, these students actually became scientists today. Some of them are still studying genetics. Right. Maybe later I can follow up on.
Okay, thank you. So, Nikki, if I may ask, now that we have these highly encouraged newcomers, what are the skills that should be targeted for a career in biocuration?
I think our youth today are quite a lot more computer literate and AI literate than we ever were. I talk for myself because not everybody's this age. But so I think the most important skills are what Frederica highlighted in the tribute to Amos and what Amos had said. It's attention to detail. It's being meticulous in your work quality. And that's just general kind of soft skills and time management. So what we found, when I was working at the EBI, and that is that if you come from a biology background, you just have a better idea, for curation about the data. You have to understand the data you're working with, because it's all very well to match things and map them to this and and try to interpret the literature. But if you don't have a biological background, I think you really are at a disadvantage. So the life sciences education, undergraduate and school education, I think is extremely important. You can look up things in a textbook, but the biology is so complex that you need to have the big picture. So I think the biggest skills you need are the biology background, understanding all the data and the organisms that you're working with, and then this meticulous nature. The need to be perfect, the need to order things, the need to pull in as much information as possible with evidence and things. So I think those are the main skills. Computing, as we heard from Alex, you can learn how to code by, with AI. So I don't think the computational skills are quite as important in the biocuration space, but that's my personal opinion.
Thank you very much. May I ask Marina, what strategies would be good to make biocuration appealing to a broader user community and for anyone who would be or decides to pursue a career in this field?
Thanks for the question. It's an exciting space, and I think you've seen over the last day and a half that our workload is not getting smaller. So that's the great thing for newcomers trying to break in. There's no shortage of amount of work. And I will say, at least within our resource, we're really starting to crowdsource a lot of our curation, which really allows newcomers to break in. I mean, like Nikki said, I think having background in the sciences will help you because just diving deep into the medical literature is difficult. But at least within the clinical genome resource, we actually take volunteers and we train them. We're very transparent about the amount of time commitment that we require. And there are different levels of time commitment. If you have very little time to commit, you just want to learn about the field, you can just annotate literature. And we have very specific tags that we use to annotate human genetic literature. And if you want to dive in deeper than that, you actually can curate for some of our panels and present to the panels, and you're trained thoroughly to do that. So I'm hoping that as we realize the scale of the problem, that more resources will do the same thing, that they'll build training programs and be able to crowdsource. If your field of interest isn't necessarily doing that at the time, I think, like Ruth said, to try to go to conferences that are relevant or different meetings that are relevant and network can be really critical, because before we started our volunteer process, that's how people joined, even as volunteers within our resources, to just meet people and then get assigned across. So I'm hoping both through volunteer work and crowdsourcing, and then also networking, that can help you break in.
Thank you very much. Peter, let's say the one identifies a resource. Oh, I like this resource a lot, and I want to be there. How do we get there?
The starting point is always, what kind of questions or data are you interested in? You know, if you are interested in human genetics, you should look into these resources, and then you can ask yourself, how can I contribute something? And you don't have to work on a database directly. You can start with Wikipedia. So just edit a couple of Wikipedia pages, you know, and make your contribution there. And you can start with just fixing typos, you know. And then once you have a bit more experience, obviously once you know your field, then you can get involved in some more specialized or even generic databases, you know. Even if you're interested in proteins, you don't have to edit all of UniProt. You can focus on your specific protein of interest. See, when I was a grad student, I worked on one protein. And actually, that was the time when I started to realize, well, there's many, many more details you can know, not only about this protein, but about the whole protein family. And suddenly you deal with hundreds or thousands of proteins. And so you have to find your niche, you know, and then you can work on that. And you have to find your community, as you said before, and you have to find the literature and the databases you can contribute to. I think that's the most, I mean, for me, it's not a technical thing. It's more like a question of what topics you're interested in. There are more the technical people who are interested in solving computational problems, and there are more the biological people who are more interested in solving the So you can find your niche no matter where it is, you know, even if it's in the medical field. So if you want to help certain patients, if you have a disease in your family. I ended up on the annual report of the Wikimedia Foundation because I happened to edit a few things on the Wikipedia page on ALS. I've not really anything to do with ALS, but one of my relatives died from ALS, and so I got interested in that. And so, even though I'm not a medical doctor, so that's one of the ways you can get involved in these things.
Thank you very much. I'm going to start with Barbara on this next question. Is what has been your career pathway, and how did you end up in biocuration or a position of influence within biocuration? And I'm going to ask everyone.
Yeah, thanks for the question. So actually that very well fits what was just said before. So I would have never considered myself a biocurator, but since I'm attending this conference, I think I am, and I have been for much longer than I thought I was. So I'm coming from the field of cheminformatics and computational drug discovery. And I've been at the EBI for 4 years now, leading a curation team. So of course, now I am a biocurator. But actually, I have been a biocurator for the past 20 years since I've been using Campbell data before that time. So I had to curate it. And I think that's really the nice thing about curation. So this amalgamation of the different disciplines. So level of detail was mentioned is really important. But I think you have to be a perfectionist as well, which isn't always a benefit, but it's a benefit for a curator. But one more thing that hasn't been mentioned, you have to really also like to document everything you do. So guidelines are very important, I think. And yeah, so for me, to answer your question, it was really more like a coincidence and the broad different expertise that I brought to the role I have now that made me a biocurator as well. Thank you.
Thank you. And may I go to your right? What is
regarding how became
how did you end up here?
well, it's a funny story. Very unexpectedly. I think it's kind of a common thing. Maybe it's many of you can relate. I was a wet lab experimentalist, fully dived into the wet lab, and kind of randomly I ended up doing a stay in the lab at the University of Padua, and they were like, "Oh, welcome. We would like you to contribute to this database. Maybe you like to curate." I didn't know what biocuration was, but I was like, "Okay, let's do it." And then I started. I was lucky enough that our group has also linked to Apicuran, which is a platform that recognizes contributions on biocuration. So they give you like scores, and it's very easy to be aware of the amount of contributions you're making, but also to show them to your supervisors. So my supervisors saw potential, my interest on it, and they wonder what I was about to do after I got my PhD, and I was like, I don't know. And they were like, would you like to get a postdoc with us to do this? Very much so. And, and this is how I am now, postdoc at the Biocomp lab and doing biocuration in this project. Yeah.
Perfect. Next, if I may ask very briefly, how did you end up here?
In
Marina.
Oh, sorry.
Yes, I'll be brief. Much like Victoria, I did my PhD in the wet lab. I was doing disease research, and so I also kind of happened upon it. I was very interested in a career in laboratory genetics. So eventually I did end up doing a clinical molecular genetics fellowship at Harvard. But before that, I didn't realize that I had to do biocuration to be in that field, because you're doing sequencing and you're generating so much sequencing data that you have to make sense of it, and you have to make sense of it in a standardized way. So I found myself in a postdoctoral fellowship with Heidi Rehm, curating data and being a variant scientist, which if you had asked me a year before that, I'd had no idea what a variant scientist was. And I feel like the field of variant science has really exploded in the last 10 to 12 years since I became one. And then it just really aligned nicely with the clinical lab too, because in a clinical system, at least in the US, you have to heavily document and validate and have standard operating procedures for everything, because you're working with patients and data with patients. So years later, I found myself directing the biocuration core and signing out genomes as a laboratory geneticist and reviewing all of the biocuration. So it kind of happened here by accident too, but I love it.
Thank you, Peter. Would you like to add anything on how you end up here? Would you like to add anything on your experience of how you ended up here?
Yeah, I mean, as I said before, I mean, I started my first database when I was a grad student, and the reason was simply there was no database, you know. And I started two databases in my life. One was on reptiles, and one was on proteins, so totally different things. But in both cases, there was no proper database in that area. And so obviously you should not start a database when there are like already 10 around. And there's like so many databases now, we kind of lose track of what is out there. But, you know, that's the way I started out, you know. And so you can always start with a small thing. I usually mention this one case which you may know, that there used to be a database on p53 mutations, just a database for one protein and its mutation. I thought that's kind of insane, you know. If you start a database on every single cancer protein, then we have balkanization and nobody can find anything anymore. So you have to find the right balance between combining data and specializing on certain areas. And so you have to find your niche, and you have to basically do it well enough. You have to focus on that one niche so you're so good that you are literally the only resource people want to go to. And that's basically what I did. I mean, I did the reptile database stuff because I was interested in reptiles as a child. And as I said, it was, you know, in the 90s when I was a grad student, there was no database. And so I thought, we need biodiversity databases. You know, and mine was probably one of the first biodiversity databases, and now there are hundreds of them. But as long as they are coordinated, and so this is one other thing, you know, as long as you are a member of a consortium or some larger organization which coordinates all the databases, that's really important, you know, so you don't unnecessarily duplicate your efforts. And I think that's one reason why we are here. You should talk to other people working on similar resources, and we share technologies and all that stuff. And so we don't duplicate and waste time.
Thank you very much. How did you end up here?
Honeybees made me do it. So as a graduate student, I had been generating a lot of genomic resources for Hymenoptera, so bees, wasps, and sawflies. And I really took an interest in figuring out how to organize my own data once I was trying to write my dissertation. From there, I went to a computational genomics lab that worked with the databases for the honeybee, the cow, genome, and other agriculturally important organisms. And then I ended up in Japan for a conference to talk about these databases. And then Pascal Godet took a look at me and said, "Get over here, biocuration person." And I've been here ever since. And it's just a journey of an obsession with organizing knowledge. And I did it with common beans when I was an undergrad research geneticist. I did it with arthropods later on. And then the last decade or so, I've been dedicated to organizing knowledge about human biomedical data. And through all of that, biocuration has been the underlying theme.
Thank you very much. Nikki?
So I did my PhD in molecular biology, and then I applied for postdocs in the UK, and then this position at EBI. But actually, by that point, I was quite happy to hang up my pipette and leave the laboratory. So I took a position at EBI. There was a Swiss procurator, and then after about 6 months, the Interpro was just starting out, and we needed. Rolf Abweiler just said there were two of us that we knew. They said, "Off you go, integrate like 2,000 entries in 3 weeks." And so we just curated and curated and curated. And actually, I didn't stay in the field very long because then I ended up being team leader of EBI, managing all the curators and all the production and all the developers and everything. But I've also had this obsession about organizing things. Ask my group, everything needs to be in neat little boxes. They will know. And so even now, you know, as I'm leading projects that have curation, I feel the need to have things organized. Trying to work with heterogeneous data just makes me realize that you really need that order. You need the curation of the data. And it's those sorts of things that, and the frustrations of doing that have kind of pushed me to keep building these resources that order things, order data and make them more useful.
Thank you very much, Gabriel. May I ask, how did you end up having influence on younger choosing that field?
Okay, firstly, I need to say that I always had an interest in genetics, DNA and genetics, all these years. I used to be a gatecrasher at workshops, even though I was not invited. That time in the 1990s, I would take public transport, travel for up to 100 kilometers to attend a workshop on a Saturday. And I think it kept me in good stead because eventually I got to meet Dr. Victoria Nibawari, and I think that basically changed my work forever because I would always be in contact with the Victoria. What can we do different? What can we do for the learners? And for me, having that collaboration, having that transition from high school to university, that was always lacking because learners would basically just study to finish matric. So exit certificate, go to university, and then they would struggle. And now having these relationships with HCIs, more specifically with Professor next to me, and also Dr. Vicky Nembawari, we were able to engage with these learners, getting them excited to follow and pursue careers into sciences. And more specifically, yesterday, guys, I don't think you guys have any idea what yesterday did for those 14, 15-year-olds. I had phone calls yesterday, and they told me, sir, we really appreciate it to be here. Teachers send me WhatsApp messages late at night saying how lovely time they had here, and they could see the effect it had on those learners. So that is what is keeping me excited. I might not be in the field like you, but whatever information we're getting, we are able to work with that in our situation, and also bridging the gap that exists between the schools and the HEIs, the higher education institutes.
Thank you very much. I'm here for the coffee. Monica, I know we've had this conversation all throughout the conference and many years before, and for potentially many years after. We're still going to have this conversation, but can you briefly touch base on how is AI changing the biocurators' work?
Thank you, Patricia. I think that Alex gave a fantastic introduction to this yesterday, and it's definitely a foundation to understanding artificial intelligence is evolving biocuration in every aspect. But in addition to the reason why it's evolving it is because we need to have a lot better structured data sets and a lot better structured information so that these machines have better luck with making predictions about the data that we're generating. And so metadata structures, file structures, of course, all of the organization of the knowledge that the biocurator generates has to be refined, or you're, as biocurators, we're now being engineers to refine this knowledge so that machine learning and our algorithms have a better chance at making more accurate predictions that are relying on the underlying biology. As we require more data and as we receive more data, better processes for refining and curating the data are going to be necessary. And as curators, we are evolving in that scalability and that capacity. I think I very much agree with the sentiment. You'll hear me talking about this tomorrow, with the sentiment that this is not about artificial intelligence replacing the job of the curator, but rather augmenting it, empowering the curator to focus on the things that make that data more robustly generated, ethical principles observed, metadata well structured, and all of these refinements that can be done to improve it. So I think it's just evolving the way in which we interact with our data and the way in which we are empowering our downstream stakeholders to draw better insights and draw better conclusions.
Thank you very much. Nikki, and I'm sitting here because I know a bit about the subject, but can you please tell us about biocurator profiles on established competency frameworks?
Yes, I've worked with Patsy and many others in the community on the ISC, the International Society for Computational Biology. They have a competency framework for understanding how you train in bioinformatics, what kind of skills, competencies, and knowledge that you need to impart when you're trying to train people in the bioinformatics space. And in doing that, we actually developed several different persona, and one of those persona was the biocurator. So it gives you an idea of the list of competencies a biocurator would need in their job across the spectrum from biological knowledge to computational skills. So that's a good start, I think, for if you're trying to work out, would I be a good curator, and what skills do I need to gain to get to that, so we can, build a learning path for biocurators. And I believe that, the EBIs and others are actually enhancing that, because that's very much general bioinformatics, but there are additional skills that a curator would need and competencies that a curator would need outside of that bioinformatics competency framework. So the EBI has actually got this competency mapper where they have an interface where they include many other competencies, and you can probably draw on competencies from like core facilities, and many of the other competency frameworks to actually pull together probably a more detailed competency set, and therefore a learning path for biocurators. So I think it's a resource that people should think about if you want to become a biocurator, or if you want to improve as a biocurator, is take yourself through this competency framework.
Thank you very much. Just to let you know, we'll share the links to some of the, or hopefully all of our resources that we're mentioning here through the notes. The resource that Nikki is referring to is the Competency Hub. And I'd like to add on that end, the biocurator persona. It's one single profile that reflects a mid-career level biocurator, but there is an opportunity to extend that profile to reflect career progression. It wouldn't be the first time that we extend. We have just extended that same profile for core facility scientists, bioinformatics core facility scientists, and there's now it would be the best opportunity to extend it for biocuration. So yeah, please reach out if you are interested in that. Can I ask, I think I'm going here with Victoria. How do we keep engaging the larger community and early stage learners to become biocurators through their own experiences as educators or biocurators or decision makers?
Well, I think a big thing is recognition. I mean, science itself, I think it's very much overlooked, underfunded, and a bunch of other qualifiers that I think everybody can relate to. So I think biocuration, it's like takes it another step on that direction. So we need to highlight the work. We need to, people usually don't even know what it is. And this has probably many reasons because it's very niche, which is a cool thing. But also it's very hard to portray your work, to be recognized. So I think that if we want people to be engaged and we want to have more people coming into the field and everything, we need to set up how this work is going to be recognized, because wet labs or other kinds of researchers have publications, and for us it's a bit trickier. Of course we can publish and we can say things about our resources, but within those like updates or whatever, there are many pieces of hard work that also need to be recognized, I think. So I think in my perspective, I started as a community or curator, and recognition was a big thing for me. And it also like kind of keeps you engaged when you see like your work is being used by others. Also highlighting that, I think it's very important for people that do science, because at the end of the day, that's what we're doing, and this is what brought us here in a way. So yeah, I'm rambling, sorry. But basically, yes, have a way standardized or a baseline on which how we are going to be recognizing these things and highlighting the work that we do. And that will set the base for people to be more engaged and interested in joining us.
Thank you very much. Thank you very much. Monica, can I, let's not everything in life is happiness, sunshine, and rainbows. What would a realistic, how do you feel a realistic career pathway would look like? What challenges may face within this career path?
Can you restate that question?
Sure. What are realistic advancement paths in biocuration? So what can we expect?
That's such a great question. And I think my most honest answer is who knows? Right, things are evolving and the landscape is changing with regards with how do we become biocurators and how do we advance. And the truth is that biocurators may have jobs that do not exist yet today, because of how data is evolving, because of how technologies are evolving. And so be prepared to be adaptable. And I think one very good thing to prepare yourself with is, in addition, have a very good foundational knowledge of the domain that you are most interested in. Your expertise in this is going to be critical. And in addition to whatever domain of expertise you decide to explore, make sure that you build some capacity regarding the at least implementation and leveraging of artificial intelligence tools to support how you do your work. If you wish to, like Nikki was saying earlier, you can build up some chops for programming. Maybe the AI can help you with that. But whatever you do, make sure that you train yourself to implementing these technologies so that you have possibilities for advancement. If you want to be in academia, I think that depending on the country, things are going to sound about the same in terms of how you enter the career of academia to become, go into a tenure track professorship or things of the like. But I think one thing that I have witnessed evolving in very positive ways in academia is also the possibilities to celebrating and promoting collaborations and working as a team. And so develop the skills of establishing successful collaborations and team science. I think that will give you an edge on how you advance in those careers. I don't think there's a specific formula, and I think my first statement is still true. Who knows what it's going to look like? So prepare yourself the best you can.
Thank you very much. Let's talk about barriers now that we've mentioned the matter. I'm going to ask everyone who wants to contribute here, what barriers do learners, especially under resource settings, face in accessing the career pathway in biocuration? Starting from my right, Victoria, if you want to add something or contribute.
Well, I am from Argentina. And yeah, places have like mine or Latin America, or where probably a lot of different places in the world struggle with funding, like on like national governmental goals are not always aligned with science. So I think, yeah, my other statement about how science is generally underfunded makes it difficult. So firstly, we need to encourage people to go into science. And that is a desirable career choice, and that you can do important things while you are also not starving. Basically, that's one of the major barriers, I think, to see that this is a reasonable and feasible career path. So I think many of our efforts sometimes crash into those things because they're not so much up to us. But I think communication and making science be out there and be like approachable to people be something that it's on daily basis, and you get to know and be relate to it. It could be a good way to get out there and demolish these barriers in a way.
Well, I can add that I think something that relates to many of the things that have been said here before. So building on those networks, right, and enhancing the networks for curators, given that it's a not so well-defined job role, depends very much on your background. If you consider yourself a curator or a data scientist, right, it's not always a clear case, but making sure that we have the networks to connect people from different continents, different environments, to be able to speak with each other, also share knowledge, and help each other to also promote themselves and others within their community. That would be important, I think. And this is why we are here, I believe.
Thank you.
I'll just say briefly, I think those in the biocuration community can really, I know we all have a ton of work, but try to pay it forward, honestly, because I know I mentioned casually networking, but that can be not casual at all to people. Travel is expensive. Workshops are few and far between sometimes, or conferences. Registration is also expensive. So I really try my best to do as much training and do in-person travel all over the world when I can and virtual workshops to really teach and try to pay it forward so that you can make those opportunities for people throughout the world, especially because we need to make all of these connections. So I think it's really a bit of an onus on us to try to advertise our field and really, if people are interested, to get them connected into the field as much as we can, even though it does take time.
Peter?
Yeah, I just have one quick thought about that because the odd thing about biocuration is that everybody wants to use data, but very few people want to take the work, and actually curate all that stuff from the literature. And so we have to bridge that gap between like our audience and ourselves. And obviously we are all interested in our data ourselves, but I think there's a lot of people outside who like to use our data. And I don't have a good answer to how we can, convince and make people actually contribute their stuff. And that's one thing we should talk a bit more about, maybe also outside this session here. But for my database, for example, I have like a mailing list of 5,000 people. And so I know like a lot of people use that stuff. And if I ask people to send me data, I do get bits and pieces of data, of course, but it's not really well structured. So you have to put a lot of work into organizing all this material. And so we need better ways to, as I say, not only pay forward, but also, get the feedback. And I think that's true for any data resource. If you work for like a big database like Uniport, I have no idea how much feedback they get. Maybe you guys can tell me, but I email Uniport quite regularly and tell them, to, correct this or that, and I don't know how many people do that. How many? That's about the same number of emails I get from my data, and that's much smaller than Uniprot. So,
Monica?
I'll tell you something that has worked for me. Sell it as a game, make him addicted to it, and then make sure that you train them so that they do it well. So one thing that I did for a long time, I worked with both people who were into arthropod genomics, but I also worked with ornithologists. And for about a good 5 years, I convinced them that there was nothing better in the world than to make sure that they curated the gene structures and for the genes that they were interested in studying. And these were people who were long careers of 30 years of looking at the same leg, so the same observation point for these birds to see how they made it, right? Like these are ecologists out in the field who have no comprehension that all of this knowledge could actually contribute to curating a database of genes for them. And so we sold it as a game for them. Here's a curation tool that you can use, and I can train you on how to do it. And next thing you know, we had people who were not otherwise engaged, super interested in making sure that they could contribute this knowledge. Field biologists, arthropod biologists as well, confessed to me saying, wow, this is so addictive. I love it. I just sit down on Friday afternoon and do some curation for your database. So just entice them and then empower them, make sure that the data is of good quality.
Thank you very much, Nikki. I'm going to skip you in purpose, but I do have a question right after that. Gabriel, would you say something on the barriers that learners face for choosing?
Yes, just to give you guys some sort of background, when I started off teaching, I was at a school where my biggest class was 11 learners. That was perfect, right? As a teacher back in the day, in the early 1990s. And then I did one of the stupid things, as some people would say. I went to teach in the township, your impoverished areas, and I had a class of 70 learners. I couldn't get to the other side of the boat to write something on the boat. But I think that was the best experience for me because I could actually see the struggles, the barriers the learners would have, and I would have to go out of my way to try to overcome those barriers. And even today, in the position I am now, I'm grateful for what I went through in the earlier stages of my career. What Professor Mulder said earlier about the barriers we have, like computer connectivity. Now, some people agree that love makes the world go round, but you also need money to go around the world. So in that case, we do like some finances. to do certain things with the learners. As you know, a lot of these things, you need funding even to get the learners here. Yesterday, we had to get finances from somebody. Somebody had to sponsor us. So for those things, we do need finances to help out with the learners. But very importantly here is that since I was working with these big classroom sizes, I got the idea of how to interact with the learners and also how to do some fun things within that classroom. However, later on, I decided to look. Let me throughout the web, that we search far and wide and see who else can help us. And here we had HCIs who came in, especially UCT, and said, look, we will basically try to make it easy for yourself. So they bring in some resources and stuff. And as you guys know, that at many schools we have a lack of laboratories, a lack of resources. And like I said earlier, with the internet, it's difficult to get these data sets, to interact with them and make it available to the learners. Even like today, I'm having an online class. And some of the grade 12 learners, they will not be able to attend the online classes because they don't have internet access at home after 3 o'clock. And in the same way, to pursue biocuration, these learners will need access to most of those things.
Thank you very much. And I'll get back to you, Nikki, with another question. Now that we're talking about the barriers for learners to pursue a career in biocuration, I would also ask what challenges are specific to biocuration project funding?
So I think in Africa, we don't have, unlike, the EBI and many other places, we actually don't have that many biodata resources, which are the biggest kind of employers of biocurators. And even if we did, they're not really funded sustainably. And so we have, that huge challenge, and then also the recognition of this as a career. So I've been working with the Global Biodata Coalition, who's looking at sustainable funding in all of the core biodata resources around the world, because I think there's a recognition for that there is some overlap and potentially some redundancy, and then there's overlap in there's no coordination in who is actually funding them, and there might be duplicate funding and then underfunding in other areas. So I think in general, these biodata resources obviously need more funding, but it's also the recognition, I think it's been mentioned a few times, oh, well, AI is going to take over biocuration, but the recognition that part of the resource is so key. If you don't have high quality data, you can't run models, and then, you actually can't use the data after that. So we need to be able to channel the funding, need to know where to channel the funding to make sure that the biocuration side of things is fully supported. And maybe there's no, try to reduce as much duplication as possible, try to refine things, refine methods so that it is as kind of streamlined as possible. But I think I was in Interpro for 7-8 years, trying to find sustainable funding for a resource, for the curators, for the developers, was extremely hard because funders want to fund cool research. They don't want to fund the core underlying infrastructure that research totally relies on. And so things like organizations like the GBC are trying to work out, well, how do you fund that infrastructure? It's not sexy to fund infrastructure.
Thank you. You mentioned something about the Global Biodata Coalition. What can you tell us about?
Yeah, so Guy he's not in the room now, but he is here from the Global Biodata Coalition. It's an organization that was set up to look at this funding crisis, really, and look at potentially sustainable funding for core biodata resources. So they've actually selected through a proper process, a set of core biodata resources that now work together and actually have these fora for these resources to discuss issues around curation, around production and that. So Guy's actually going to be talking tomorrow, so he will hopefully tell you a bit more about it. But I think it's a really good step forward as having a coordinated body to approach funders, to get the funders to work in a coordinated way to make sure that all the necessary resources are funded long term and that there are no major ones sort of falling through the gap and others being sort of double funded.
Thank you very much. With this question, we are expecting guide dogs for tomorrow. And we are opening the Q&A for this panel. So if you have any questions in the audience.
Thanks a lot. And I was just curious if you had a comment, and it is open to the whole panel, around the professionalization of biocurators, and in the context of universities, which are very rigid in terms of how they're structured. You're either an admin person, you support staff, or you're academia. And I was wondering if you had experience in how do you engage universities for the awareness of a category of staff which might fall through the cracks. And in the early days of bioinformatics in our country, we would hide somebody as a web developer. So you'd had 10 web developers, but they did no web developing. But that was as a way to assimilate them into an ecosystem that was quite rigid.
So I can maybe start. I don't have a solution because I'm in the same scenario as you are with these academic settings. You know, when you have a curator, data steward in a university, they're a librarian, and they're not working on a particular resource. I think these new initiatives like so we'll hear about the African Open Science Platform tomorrow, these new open science policies. So South Africa's just passed their open science policy, and then the implementation plan. In order to have an open science ecosystem at an institution, you have to have things like curators. And in the implementation plan, there is a discussion around actually trying to recognize these as bona fide careers and positions within universities. But you're right. Right now, we have to be creative, and you can only do it on external funding. You certainly can't get an internal UCT post in biocuration. But hopefully, as more of the open science kind of policy implementations come along, the universities will be top-down pushed into recognizing these careers that are necessary to enable open science.
Hi there, my name is Tony from University of Cape Town. It's really good to be here and hearing the different perspective from the panels. So my question to Peter, we have all seen top-down good government policies really play an important role. And we've all seen this firsthand during the SARS-CoV-2 pandemic. But on the other hand, when we look through a different lens, we can't omit, or we must all acknowledge the role that local community play in early disease detection and prevention. So when we look through these two very different lenses at the same problem, how do we start integrating the different data standards or the different biocuration policies that we have spoken about? And perhaps a second question, if I may, to Professor Nikimoda. Thank you very much for your presentation. And I think from that, we've all seen this training gap or skills need. I mean, close to 1,000 applications, we will have 30 spaces, if I remember correctly. And I mean, I look around the room, there's professors, doctors, you're all at the top of your research field. And to be honest, you are the custodians of knowledge, opportunity, and funding. How do we ensure that we disseminate equitable training, to all levels of society? So someone who's doing the tertiary education versus a girl in a rural township doing their primary or secondary education, so we ensure that no child gets left behind. Thank you.
So the one question I heard was that you asked about data integration and standards and stuff. Is that correct? That's almost a bottomless pit question, so I'm not even sure how to address that, because so we have seen a bunch of talks here at this meeting about data standards and attempts to standardize and integrate data. I think the problem is, I know of several bodies who develop standards, but the question is, do people really adopt them? You can have the best standard in the world, but if nobody uses them or if they are too complicated, I mean, they will not be successful. So I think you have to find the sweet spot of having standards and usability. And I think that's the key question. Each of you guys have to, answer that, or everybody of you guys has to answer this question for themselves. are the standards you're using or you would like to use, are they user-friendly enough to be really implemented, or are they just too complicated? I mean, I can only speak from my own experience. As long as your database is successful and people use them, you can be fairly sure that you have at least decent standards. And if your resource is not used a whole lot, probably the standards are too complicated or the user friendliness is not there and all these things. I cannot comment on, you talk about COVID and stuff, so that's a different issue. But do you have any comments on this?
I have a lot of opinions about it, and I agree with you. It's a very, as we look through all of the talks of the past 2 days, it is incredible for me to see. Look at us. We're all trying to solve the same problem, right? This is essentially the same underlying problem, and some of us have managed to do it with more luck than others because the community has adopted this. But my question is always the same, right? Like, are people capable of adhering to these standards, how easy it is? And so the best answer that I have been able to come up with in order to help adoption that is widespread, that is easy, and that is registered is to join a standard body that helps you with this, because it brings together community. So Global Alliance for Genomics and Health, where both Nikki and I are members, is one of those attempts. We are trying to standardize the way in which we represent it, and we try to bring a large body of researchers across the world behind it so that we have both contributions, stakeholders, policy checks, ethical checks, so that at the very least we have something that a large body a large portion of the research community can stand behind.
Thanks.
This is about careers in biocuration. I guess one of the greatest challenges for having a biocuration career is the questions you might ask them at interview. So I wonder if anyone has something, sort of an interesting question that they like that they ask at interviews, and then maybe people could have a think about that.
I've just one quick. So you're asking about the questions I would ask a candidate. I mean, I guess again the key question is, can a candidate bring together enough enthusiasm and interest in a topic with the technical skills to implement stuff? So domain knowledge as well as some computational knowledge, like some scripting skills, or maybe these days just enough skills to prompt Claude to write some code for you. I think that would be super helpful. Any other comments?
I often ask about like communication skills. I feel like people often are just thinking like curators hide under a desk and just type all day. But as we know, it's like really big team science at this point. So I feel like they need to have some sort of way that, how do you work in a team? Where do you see yourself working in a team? And how do you communicate? How do you write? How do you present? Just so that you can also really sell your work while you're doing it.
I can add one sentence here, because it's like for any other product you try to produce, you have to satisfy your customer needs. That's the most important thing. So if your customer, your audience does not use or like your resource, you are basically,
Quite frankly, I don't think there's anything revolutionary about this, but something that I really, really like to ask that helps me get a little bit of a better idea of where this person's enthusiasm is and their objectives. I literally asked, what makes you happy about the science that you do? And then from there, I have gotten really good perspectives.
Okay, so I think we are standing between everybody's lunch. So it's time for ending this panel. Thank you very much to everyone who contributed, into the audience who was here to talk with us. So thank you.
Thank you.
Apologies, there's a talk.
Thank you. So now we will go forward to our last keynote. Professor Alan Christopher from the University of Western Cape, South Africa, and which will provide us with his talk, Data Curation Across Africa's Infection Disease Landscape. Professor Alan Christopher, welcome on the floor. So Professor Alan Christopher is a South African bioinformatician, bioinformatics scientist and academic who serves as the director of the South African National Bioinformatics Institute at the University of the Western Cape, and as director of the South African MRC Bioinformatics Unit. He holds as a DSA NRF Research Chair in Bioinformatics and Health Genomics, and he is internationally recognized for his leadership in pathogen genomics, infectious disease research, and computational analysis of high-throughput sequencing data. His work focuses on developing tools and methods for analyzing genomics data in public health, including platforms like Comba, TB, and BioBab. And he has played a central role in strengthening genomic surveillance and data system across Africa, including senior advisor roles with the Africa CDC and leading the Public Health Alliance for Genomics Epidemiology. Professor Allen, please, the floor is yours.
I appreciate the invitation to speak with you today. And I know that I'm definitely standing between you and lunch. And I think that maybe what I want to do is what I want to take some time now is maybe just to shed a spotlight on, I think, a scientific discipline that could certainly value and benefit from a closer relationship with the biocuration community. And by no means am I adding additional work on your end, but I think for those of us who are engaged in the collaborations in the public health space, I think it's a reminder that we need to somehow find ways in which we bring these two areas closer together. And so maybe just in preparing where I'm going, want maybe just to touch on public health implementation and research, maybe contextualize the need for standardization in these public health laboratories, and then think through how do we sustain curation within the setting, given what we've even heard now around the funding frameworks and how that gets maintained. Maybe a comment about the use cases, which you've heard a lot about yesterday afternoon, and then time permitting, I can move on. So let me just start with my conclusion. So if you need to run out for lunch or you maybe doze off, I think there is a unique opportunity to push public health laboratories into the curation space, particularly now when we have seen in the last 5 years, certainly through the COVID pandemic, where resources have been mobilized in Africa to strengthen national public health facilities, where they really represent a legal mandate nationally to drive a national response to a disease threat. And so when we started to work alongside public health institutions, we realized that this is the tension, right? You've got research, inquiry, research in knowledge production, but really it is how does that go full circle in order to inform a public health response or inform a public health policy? And I like the talk by Sebastian yesterday on microRNAs. We made the side comment around knowledge production with application. And this is really where we are at. And so when one is looking even at funding models where it is knowledge discovery, it's difficult sometimes to engage funders in the implementation science that embeds the knowledge production into a public health space. So this represents somewhat the workflows in these national labs. They're very much driven by disease surveillance. And yesterday afternoon, my colleagues Dominic and Eddie
spoke really about the standardization or the managing or the organization of data as it enters into this workflow. And I think for us thinking through how do we move these public health facilities whose mandate is in fact health service delivery, how do we think through changing a culture in there where we potentially could embed curation along this entire workflow? And so where it all started, we've seen the migration and the growth of national public health labs to embed genomics within their offering. And we've certainly seen in Europe a wonderful assessment done in 2016 already, where the value of genomics data informing some of the diagnostic approaches that are being used and how that had a positive impact on patient care. And so we saw now in the last 5 years, a lot of it channeled through the Africa Center for Disease Control and the WHO in mobilizing resources to these national facilities. And this is an example more of the infrastructure resources that have been mobilized. But I mentioned in brackets there the workforce strengthening. And so what we saw with a strategic response during COVID, the need to strengthen laboratory infrastructure in response to the COVID pandemic, recognizing that while we strengthen infrastructure, you are building national capacity for data production. And at some point along the plan, you will have to manage the data that is actually produced within that. And so we've seen, just in terms of numbers, you've seen this plethora of workforce that is engaged at public health response. And so then the question we are often answering, to what extent could we engage these personnel in not only as a service delivery in public health, but also as a way of thinking about the research component? And so let me hastily say that there are certainly national public health facilities which manage to accommodate both research to inform the applications within public health, but for many countries on the continent where the national public health labs are at the sort of at the early phases, there's a lot of reliance on neighboring academic institutions in order to help support the provision of applications that can be applied within a public health setting, which very often is very regulated because of the need to accredit these processes. And so we have seen on the continent a shift to make sure that every national public health lab has the wherewithal to stand up the appropriate wet lab support. And so the idea of ISO accreditation and the processes involved in that. And so as that is progressing, what I think as a community, as a bioinformatics community, and by extension, the biocuration community, we have to ensure that in addition to the evolution of laboratory standards in our national labs, that we also facilitate the quality assessment of the bioinformatics data, which is coming through these labs at an alarming rate, in fact. And so we have a push towards thinking through what does the ISO for bioinformatics look like? And maybe just a plug for some Wellcome Trust-funded project. This ISO in a Box project, which is actually led by Tom Connor at Wales in Cardiff, is the idea of how do you build the appropriate infrastructure and tools that could move a public health laboratory to have the capability of verifying, validating, and providing the appropriate data from your bioinformatics analyses that could inform a clinical decision. And we've seen some of this in more recently coming out of Australia. We're really bringing some of the genomics pipelines that are being used in a research context into the applications to health. And I think more and more of this needs to be done. And I think the question that is going through a lot of our minds is when we look at the workflow within these national labs, to what extent do we embed a curation mindset in underpinning the development or the standardization of both the data and the tools that could then lead to clinical application? I've gone through that. And so I think this is where our partnership with Africa CDC has been quite useful. And we've seen certainly in an African context, the activities within this regional body in supporting countries in the detection, the characterization, as well as reporting on various disease outbreaks. And we've listed just a few there. And I think the point really here is that as a continent, there is about 130 diseases that are monitored annually. And so many of them do not become diseases of international concern, but they're certainly of concern regionally. And so could these be the segue into strengthening curation within this particular scientific domain? And I think one could whittle it down further, and certainly this has been the strategy used on the continent in the public health space, is to think through what are the use cases that could be the entry point for standing up the appropriate genomic support, and in the context of this conference, I think, stand up the type of curation and the standardization which has come through a lot of the talks that has happened this past two days. And so these really become the strategic plan for what is happening via the Africa CDC, and really boxing these diseases into certain categories and trying to respond appropriately. And so there's been a number of implementation plans, and I mentioned this purely that if you want a good sense as to where national and regional emphasis are moving, I think these activities and these documents become a useful one. And over the last 2 years, and maybe 2.5 years now, public health practitioners, genomics scientists, bioinformatics scientists have come together and trying to formulate a guidance document for those officials that are trying to raise funding in this space, to try and give a tangible examples of how we could respond to antimalarial resistance, antimicrobial resistance, actually, malaria and the vaccine preventable diseases. And these documents have been generated, and the last of which was released probably about a month ago now. But they provide a good footprint as to where things are heading. And so I think for me it is to remind you again that the need for the curation in the pathogen space, certainly in Africa, is well placed given how national public health labs have been resourced over the last few years, and how that mobilization of resources has resulted in increased local production of pathogen data. And I've made this point in a previous slide, is that the quality systems that have to evolve alongside the laboratory quality assessments which are happening. And so I think that for public health laboratories whose mandate is to be the first response to disease outbreaks, how do we as a community agree or get a consensus, find a middle ground in standardizing processes in times of peace, so that when the crisis occurs, there is an understanding of protocol and also of what gets collected in the field, for example. And so that is really what we see as a primary need within the public health labs at the moment. And so with that in mind, the Public Health Alliance for Genomic Epidemiology, phage, partnered with the Africa CDC and really trying to see, could we approach that initial part of data collection? And so I just put here, for those of you that are not familiar with phage, there's a number of working groups, and this is an international consortium, really where the heavy lifting and the engagement is happening in trying to formulate standards or guidance in various areas. And so the work that you've heard yesterday afternoon by both Dominic and Eddie has certainly been work that's happened in the context of data standards. We have a team looking at pipelines and guidance documents in how do you actually provide assistance to people in shaping how they do various analyses. And so I think maybe the next two slides is maybe just to alert you to, I think, potential drivers of curation. And I mentioned to you some of the priority pathogens that are forming the key points of mobilizing funding as well as operationalizing these things. And I think these become good drivers for where one could try and bring the public health community closer to this curation space. And one of it is in the cholera. And so there are about 7 countries involved in a cholera network together with some international scientists, and really looking at both the collection of material locally, but also monitoring the transmission dynamics within the region, as well as identifying certain antimicrobial resistance markers in the space. But I think what I wanted to point out particularly for us as researchers is that we have to get our heads around the fact that there is certainly a data collection component, there is a processing and analysis, and the outputs of that is not a student thesis or a manuscript per se, but it has to translate into some policy component so that the decision makers could then use it appropriately as they respond to a particular disease threat. Similarly, in the malaria space, and Eddie spoke about this in the last talk yesterday, we've certainly seen the collection of biospecimens around the region, and analysis of genetic markers within this context. Not only WHO endorsed genetic markers in the Plasmodium, but we've also seen a number of other mutations that have been highlighted, and that will require obviously further work. And what we're really asking as a community is, what does that mean in terms of policy and standardizing the data to inform what your national malaria control programs have to work with? And so I'll put in about 2 slides on this space, and that is, as a community, both the malaria scientists on the continent and off the continent came together to really understand what does the data look like at various layers. And this is just a quick snapshot that our data at the clinical space, some of that is very much hidden because of the regulatory requirements within hospitals. There's epidemiological data, which more and more is finding its way into this DHIS2 format. There is a biospecimen collection component. There's genomics data that's being produced. And countries where malaria is endemic have these national malaria control programs. And so the genomics data that is coming out of your laboratories are being passed on to these malaria control programs, and the mode of that transfer and the interpretation of that data is in fact a challenge. And so if you start to interrogate the mode of sharing the genomics data with your national malaria control programs, you find that that sometimes is provided in an email, as a PowerPoint slide, as a spreadsheet that has far too many rows in it. And so the communicating the data from the genomics lab to your control program where the decision-making has to happen requires a rethink. And once you, as a control program, in fact, see the data, what informs the final decision? And so that is something that we have been looking at as a consortium. And so this just shows you some of the thinking around. There are a number of markers, and you could stratify that by its relevance in surveillance. Down here are markers in the context of the vector for malaria. And as you're going up, you're looking at some of the drug resistance mutations that reflects the diversity in Plasmodium. And so with that, the teams have actually looked at a minimal data set, and we've heard a number of speakers speak about what constitute that core variables that has to be captured so that when you pass that on to the decision maker, you are honing in their attention to things that actually matter. And they really don't care about a VCF file, a FOSQ file of documents. And so this has really been around the collection of biospecimens, the laboratory processing, the analysis, and then the sense making of that knowledge, because the decision maker needs to have something within their hands in order to actually communicate to the authorities. And so what we've done is we've had a number of work streams looking at this and what does the genetic markers mean in the context of treatment? What does it mean in the context of initiating a particular public health response? And so the work that has been mentioned yesterday afternoon by my colleagues has been put up on these resources. Initially, when we started out this, the data standards within the data standards working group, which has been headed up by Emma Griffiths. A lot of that has also evolved into the work that has been done in the context of malaria and cholera, as well as neonatal sepsis. And so if you do want to go have a look at some of the specs that have been developed, please feel free to do that. We have embarked on a few newer projects in the context of meningitis, which I think is an exciting continuation and exploration of how do we formulate that engagement among a research community and a community that's involved in implementation science. And so that what comes out of that engagement informs public health decision making. I told myself I will check my watch at this point to see where we are. Tell me how much I'm going to talk about this. So I've got about 3 minutes or so. And maybe just to say that there are a range of projects coordinated regionally, and I've spoken about one component thereof. And a lot of the questions at a national level in countries is, how does this data get communicated across borders in the midst of a crisis? And so very often data, in fact, does not cross borders quick enough in order for countries to collaborate on responding and for countries to know in advance what potentially could be happening. And so there was a project that started about 6 or 7 years ago now, really trying to see how do we collect, and as we spoke about this past 2 days, collect, organize, box in, figure out how to make sense of placing data in a way that can be shared easily, shared fast, and also reproducible. And as my colleague has mentioned earlier, is that you're also dealing with some contextual dynamics which have to be provided for. And so this Agari platform was built. And Agari is an Amharic word that means to share and also gain value from. And essentially what this is allowing national facilities to have a snapshot of the genomic data that they have access to. It allows them to keep a handle on their national projects that is driving their legal mandate, as well as the support that they get from academia and the ability to then initiate new projects. And not only initiate new projects, but also share that so that it's a collective response to the crisis. And so putting into the hands and giving countries the agency with which to manage that collaborative response. So where's this going? Your lunch is nearly there. So this is really my concluding slide. I think what I'm wanting to communicate to you is that, in addition to the different domains in which we operate, there is a scientific domain in the health space, not just the clinical space, but in the public health surveillance space, which is highly regulated. But there's an opportunity to influence how data is managed within that space and to facilitate and also communicate the value of ensuring the quality of the data that goes in there so that you guarantee the utility of that in evidence-based decisions. I've spoken about the fact that the use cases often in this example is the application to a disease outbreak. And that is really the wrong time in order to engage in curation. The chaos that surrounds that particular time period is in fact not the right time. And so that is, for me, it is how do we in our engagements with public health bring them closer to biocuration when there's no crisis? Because that, I think, is when you start to engage people, encourage a different culture of working, a culture of ensuring the accuracy of data that is being produced at large volumes, facilitating the cross-border collaboration, and obviously the impact on disease transmission. And I think you're wanting to really facilitate a wider sharing of pathogen data so that we can deal with these health crises that is obviously we have not been spared globally. And so with that, maybe just to wrap up and say that there's been a number of funders, a number of people involved, and I just want to thank those that have been involved in this and recognizing that these are partnerships that will facilitate the ability to respond both locally as well as globally. Thank you very much.
Thank Prof. Allen for your informative talk. And now we will move to the audience questions, if any. Okay, so I think it's
time for lunch.
Yes, thanks for lunch. Nikki, yes, please go ahead, Nikki.
Thanks. Great talk. So the ISO kind of accreditation, who would oversee that? It's not, it's a really good idea because in public health, as you say, everything else has got accreditation, but data and bioinformatics component hasn't got that. How do you do the assessment and who's the accrediting? Is it the ISO still, or is it kind of more Africa CDC accreditation? And is there something like that elsewhere in the world?
So the ISO framework has certainly introduced ICT components to that. And so I think over time that will expand further. And oh, sorry. Oh, apology for that. So yeah, so I think at the moment the ISO standards are evolving to incorporate a genomics component of that. I think we're at a space now which I think is so fluid. Normally it will be an ISO body that would deal with that. I think the approach a number of us are taking is that in the absence of the cost and the frameworks that are available there, could one look at an idea of being ISO ready? And so the project that I alluded to that is funded by the Wellcome Trust is an attempt to put together computational methodologies that will facilitate a laboratory to be ready for an ISO standard when it does become available. And very often these standards are about carefully tracking what is being done in, this context, the life cycle of the data, making sure that there is a trail or a footprint of the resources that are exclusively used in the context of the clinical space. And so in our research spaces, there's one server that is used, you name it, for plants, for human, for anything else. In the context of an ISO accredited space, you have to guarantee the exclusive use of a resource. And so putting together these tools is something that is shaping this ISO in a box, which is now going by the name Adviso. I think in addition to the tools, very often when you're wanting to accredit a process in the laboratory, you have to have a data set which is regarded as the gold standard. And so when you're talking about whole genome sequence data, what becomes the reference standard? And so in the paper that I showed you from Australia, it is, can we put together those validation data sets that people can utilize in order to test and approve their processes. And so I think that is where it's going, but that's a long answer to your question around who will actually oversee that. Currently, it is not being, people are taking the responsibility of wanting to become ISO ready. And so that is exactly where it is at. And so there's not one body that is currently doing that. Yeah.
Any other question?
So unless somebody else wants, just a quick question about the biocuration side of things. So in public health, in academia, we just discussed it was difficult to create these positions. In public health, I think in some of the institutions, there's so little resources that one person's doing everything. So a lot of the biocuration is actually left to the bioinformatician who's developing the workflows, implementing them, and that. Have you seen in the public health institutes actual positions just for a curator, or is that still not a reality?
That's a great question. So no, it's not a reality. And I mentioned early in my talk, we have national laboratories at various stages of development and expertise. And so certainly within that, the spaces where they certainly provide a research component in genomics to support their laboratory services, those positions are also not specifically curation. When I think, which is why I asked the question about professionalization of this position, it is to what extent do we in our specific environments start to agitate and promote the value of and the development of this particular position and job description so that the people that are actually involved in this become aware, because there's no human resource people in this room, right? And those are the first people who come and tell us, I don't know, what's a software programmer? Do they work for our bachelor's degree program? And so I think, and the same I think is in the case of biocuration, we don't have them certainly at my institution, and I mean the same at yours. And so yes, in the public health space, even less so. And so as we start to engage with decision makers, I think it's just creating the awareness of, and hence I think I like the idea about how does the research community as well as a community that's involved in implementation services actually work alongside each other to actually see the value add for these specific job roles.
Great talk. So I just have one question about surveillance is happening at a national level, maybe public health agencies. And I know NICD has done a great job in genomic capacity. Do you think this expertise will trickle down into hospital and to improve diagnostic, for example, using metagenomics?
Yes, so I think I'll have to speak from the context of my own country. So the way that the hospitals would do is they would request services or diagnostics to the National Health Laboratory Service, and then that results would feed back to the hospitals. And so I suppose the question for us would be, will we see the application and the expansion of that kind of technologies at that National Laboratory Services? And I think this is probably a two-edged sword. The one is you see the value. The other thing is how you have to actually change sometimes an entire ecosystem in order to adopt new methods. So we've done something a certain way, it works a certain way, but the health economic impact of that methodology will have to be demonstrated in order for people to say, well, we will now invest appropriately. We've seen that change coming in with whole genome sequencing data being used for TB drug resistance, and that is slowly moving in that space, but it does require the data to inform authorities about the need to change. And I don't know to what extent that and how quick that shift will happen.
Another question?
Great talk, really interesting. I'm just curious, I saw on your last slide, and you said this, you're highly collaborative. You have many groups that you're working with together. I'm curious how a global pandemic may have changed your strategy, and if it caused additional collaboration or people throughout the world to reach out to you, because Africa really has had some amazing surveillance, and I feel like a lot of the pathogen detection has been state of the art, and many people didn't think about that necessarily before there was a global pandemic.
And so the question is, to what extent that people actually approached us as a group?
So how did your strategy change after a global pandemic or during a global pandemic? And did it increase collaboration outside of Africa, of people reaching out and, asking you how you were doing things?
Yeah, and so certainly in terms of during the pandemic, I think what was clear was the need to rapidly move from what we were doing. And I think when you're dealing with public health laboratories, which are very rigid in how they operate stuff, I think strategically is getting the correct decision makers involved. It was actually interesting to see that we constantly have a problem with supply chain. Supply chain got solved during 2021 and 2022. And supply chain became a problem again after 2024. So what was different, right? And so it was the crisis that pushed people into looking at alternative ways to respond. The difficulty with politicians are that they have very short memory spans. So things have moved on. And so certainly for us, to come back to your question then, how does it actually shift how we operate now? I think it is early engagement, one, with decision folks, making sure that they are constantly being sensitized to the need for and also the application of these tools. Certainly for colleagues of mine, for example, in Uganda, it's been amazing to see just how folks have engaged them globally to understand also how they've moved to a 2-day response in terms of isolation and, well, detection and isolation and characterization. And so I think we have seen more and more of that come on board. And our countries have stood up a public health response during an outbreak by quickly mobilizing the appropriate actors in order to actually solve that.
Thank you. Yes, so 2 questions. Thank you for the great talk. So the first question is, are there any plans or maybe discussions around incorporation of AI into your systems and platforms? And then another question is around maybe the incorporation of commercialization of some of your systems or products or the research products that are coming here, whether that has been thought of or embedded. Sorry if I missed those points.
So in the context of AI, so work that is happening within Phage certainly is around how do we utilize machine learning in order to assist with the time-consuming part of gathering the laboratory data, the field data that's coming in, and providing a narrative to the public health official. And so a project that is funded together with Nikki, project where I have a postdoc that is looking at how do we get that narrative, which they call a policy brief, how do you get that quickly to an official? So in the work that we were doing in responding to cholera in Zambia, you have information coming in from the field, you have laboratory analysis being done, you have a bioinformatics component, and that might happen on day 1. By day 4, there are more things coming in, and the amount of time it takes to manually assemble that narrative. And so what happens with these AI machines, they actually help in just trying to piece together some of that, and it gives you a draft with which you can then work with in order to prepare the communique for your public health person. And so certainly that has been something that has been a project that we're doing with Uganda Central Public Health Lab, and we foresee us trying to scale that up for different pathogens moving forward. Your second question around commercialization, not in the context of the projects that I've been doing or that I have links with, but I would imagine that certainly in the context of diagnostics, I would imagine that that would be the spaces where people are moving rapidly towards trying to have the wherewithal for early detection. And this has really been the challenge that has been faced in the response to mpox. How do you immediately identify and diagnose people, and having access to and the availability of these diagnostic kits become quite useful. And so I would guess that that would be the spaces where people are starting to look at.
So with that, we will finish our session, and with the big thanks to your contribution, Professor. Thank you.
Thank you very much, sir. Thank you. Oh, we can take a photo. Sure. You all are.
So I'll introduce you because we're really answer to go. Okay. And yes.
I have to take all of my, I can pick here my laptop and my information. I will bring it one. Okay. If I have to, no one else is giving it.
ما مش لك كنت راي فروز وكهو. Hi everyone. How are you? تبي جوست نعمل هكا هيش. انا جوست حبيت اثبت كي نرى روحي لهنا نجم نرى الوحده كيف ما نحب بالبنات ولا بليش
حقا خلينا نعمل بلاش ترى امشي ترى الو الو حول 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 امشي هاي هاي فن امشي
Hi. Keynote speakers. Hi. وجهي ضاوي مش نضلي. Hi. اليوم يا سيدك بسيدك شنعنا في السوشيال ميديا تيم شو نقولك باهي نتبسم
حقويني هذه شيري هذه نكرهها نحب الشيرة هذه ها ها يعجبني وجهي على جنب أكثر اي خاطر يعجبني وجهي أكثر دونك توا مروة خذيري تنجم تجي تقدم سبيتش نتاعها مرحبا ابلوديسي الي وي applaudissez pour Rim. Applaudissez pour...
Are you ready? Yes. Okay. I think stop. Oh, you're touching me. Oh. Okay, and maybe I need chair because I can't. Then how? That's for you. Is it possible to be this way?
Yes.
So I know what to do.
It's possible to see from here.
Okay. If she sits, she might not be able to. No, she won't be able to see. So she can actually sit here. Then they bring the mic, the detached mic.
Yeah.
Then she sits and presents. She would look from here. So you could sit here.
It would be good.
Okay, here?
Yeah.
Okay.
In here to this bench, this table. You just shift this chair. Yeah. Can I shift here?
Well, would you see? Okay. Okay.
Here.
Yeah.
That's
Here?
it.
Now I can see the screen also. And how about my mic?
The mic.
So
Oh.
Use-
You will,
So where's this?
All right. Okay.
Does that work?
Yeah, that's great. Thank you. All right.
I'm sure he'll show up. Well, guys, I was checking that lunch. You didn't see? Oh, maybe after the presentation. Yes.
Yeah, there should be a break in that now. Go back in it.
So once, yeah. Okay. No, that's fine. No worries. We're not using these cables anymore, so yeah. Oh, okay.
You think you're gonna give it that mic or this one? Or that one?
I think this one. The second and then now can just put this one a little bit.
In the microphone?
Sorry?
The microphone, you say maybe that one or not?
No, she gave me this one. Then I can push the next one. The side.
Otherwise you go like this, and go like this with it.
With this one.
Because then this one, what happens, you're gonna sit to like this. It's gonna keep it, you go like this. Whereas that one, you can pop right in front of it, you can just talk. Hey?
Also needed to focus on the, because the,
See anything on,
show on,
see anything on the notes?
there's nothing.
Just scan, get the-
Oh, okay.
the-
On the,
old school.
okay. So just move this one.
You just do like one of these stories.
Yeah.
You can do it from this side, you do it from this side, bro. You're gonna walk, yeah, come here. Get this, look like it. Down, you're gonna go like this, eh? Drop that thing right down. Put it right down. You have to go like this. Go. The other side. Good this up. Huh? She's gonna walk up this up.
Good afternoon. Welcome to this session on databases, knowledge bases, other bases, and knowledge graphs. So in this session, we are going to have an interesting session on how to organize biological data. And we have lined up interesting number of presenters, and to kickstart us off is Dr. Nivedita Yadav from the Indian Biological Data Center. She's an expert in genomics, transcriptomics, and phylogenomics. She's going to tell us about BioVerse, a regionally relevant resource in India, and how it can be applied globally. So help me to welcome Dr. Nivedita.
Imagine a nation whose biological wealth spans from peaks of Himalayas to the depth of Indian Ocean, where traditional knowledge, modern omics technologies, and computer science together create extraordinary scientific resources. That nation is India. But most of these biological resources remain scattered and underutilized. Hello and namaste everyone. I am Dr. Nivedita Yadav from Indian Biological Data Center, and today I am delighted to present IBDC itself and introduce BioVerse. First of all, thank you organizers for this opportunity and hospitality. So let's start. Before I jump to BioVerse, I would like to give a brief introduction about Indian Biological Data Center. Indian biological diversity. India is one of the most biological diverse country in the world. Yet, and these diverse data nowadays being captured through large-scale data generations across Indian institutions and national projects. Many biological data is being generated nowadays from gene to genome, from proteomics, phenotypes, images, and it comes from human, animal, plants, and all the entire ecosystem. And it is also empowering the Indian research domain from agriculture, research science, medical science, and all other domains. But with these huge data generations comes some challenges. And the challenges are how to store, how to manage, and how to share these biological data. So to address these challenges, Government of India, Department of Biotechnology established Indian Biological Data Center. So IBDC is India's first national life science repository for life science data submissions, and it is supported by Government of India through Department of Biotechnology. It enables Biotech Pride Guideline for responsible data sharing, and it is joint projects of Regional Center of Biotechnology, National Institute of Immunology, and International Center of Genetic Engineering and Biotechnology, National Informatics Center. IBDC also have data recovery site at NIC Bhubaneswar, where real-time data exchange happens. So the IBDC is located at Regional Center of Biotechnology itself, which is research and academic institutions, and it is established by Department of Biotechnology, Government of India, where it also works in the collaboration of UNESCO. So it was dedicated on November 10, 2022 to the nation by Honorable Minister of Science and Technology, Dr. Jitendra Singh. So what actually IBDC do? It mainly works in 6 fields. First is biological data archive resources, high performance computational facility, support to national projects, big data analytics, user resources, and trainings on data submission and data analysis. One of the most fascinating services of IBDC is its high performance computing facility, which is known as Brahma HPC. Brahma's HPC have a huge storage capacity of 5.5 petabyte and computing power of 961 teraflop, which means it can store millions of data and can compute large complex biological files. IBDC is also planning to expand it as 10 petabyte, and currently we have 131 users from 72 institutions they are using HPC. So this is the ecosystem of Indian Biological Data Center. So IBDC is not just a repository, it's a complete ecosystem where data submission, storage, and advanced services connects each other. So for gene, genome, and RNA, we have data repositories on Indian Nucleotide Data Archive, Indian Nucleotide Data Archive for controlled access archives, and Indian Array Data Archives. These are for gene, genome, and RNA. For protein, we have Indian Proteome Data Bank. For chemical, we have Indian Metabolome Data Archive. And for phenotypic, we have Indian Phenome Repository. And for image data files, we have Indian Biological Image Archive. On the other hand, we also give some services and application like ISDA, Indian Structural Data Archive, which is the mirror of PDB, but here we also curate Indian structures. On the end, other is IBDC Data Access Portal. Here, all the data managed access and control data, they are sensible data. People can access those data from this portal. BioNode, it's a kind of toolbox where we provide users to do their biological data analysis, and BioVerse, which I'm going to explain in the next slide, and BREM HPC. So first of all, Nucleotide Data Archive, INDA. It is basically to archive and manage nucleotide sequence data generated in India. And it also supports synchronization of synchronization within ENA and NCBI at the same time. These are the key statistics of INDA. Next, its control version. In this data repository, it is designed for sensitive nucleotide sequencing data. And we have INSACOG. INSACOG means Indian COVID catalog. And Genome India. We have also sensitive data from Indian Human Microbiome Initiatives. If we talk about Phenome repository, we have functional two portals under this umbrella. One is Indian Crop Phenome Database and Indian Animal Phenome Database. All these databases follows FAIR principles, and these are the key statistics. Even the crop phenome database is itself a unique database, and we have some major national mission mode projects on wheat, rice, chickpea, and linseed, and many more. And in upcoming, we have a human phenome data archive and microbial phenome database in the queue. So if we talk about metabolome database, we have IMDA. IMDA user can accept raw, submit raw and process metabolomic data in this portal. For image database, we have Indian Biological Image Data Archive, and it facilitates AI/ML-based data analysis and research, and it supports multimodal biological image data from microscope, MRI, and CT. If we talk about Indian Array Data Archive, we have this portal, especially for array-based data submissions. It follows Miami, and here user can submit array-based high throughput label, multi-throughput label, and low throughput label. For protein submissions, for protein, we have Indian Proteome Data Bank. It accepts mass spectrometry-based proteome workflow, and it's also have unique identification and open access to mass spectrometry-based data sets. And these are the services, as I told, that it is a kind of mirror of PDB, and it also have curated Indian origin data, structural data, record with respective meta information, and IBDC, a data access portal. It is for accessing managed biological data set with the help of feed protocol with these guidelines. And this is the toolbox. We have currently 3 modules here for biomanufacturing, genomics, and structural biology. User can come and they can use it for the analysis of their biological data. Now we talk about Bioverse. So according to research and publication, India is rising in bioinformatics and computational biology research. At the same time, it is also developing in the development of biological resources. According to Database Common, as we can see in the graph, India is leading country in the development of biological databases. But here also comes some challenges. Most of the databases and tools are scattered. They are spread across independent website, and it results in low visibility, and people with this low visibility, it would be hard to discover a specific resource. And sometimes it become access, but there is no standard protocol how to retrieve, how to use that biological resource. And it results in low reusability, and sometimes it also produce duplicated efforts. So there are some existing platform, but it comes with some gaps. Like in database common, it is only focused on biological database, and omics tool, it only focus on omics based tool, and bio tools, it again it's focused on tool and software. They don't integrate and database, and also there are some limited metadata and annotation about the tools. In NCBI, it is a broad repository for global biological data, but it is not designed for integrated tools and databases, and there is no accession system or external resources. And if we talk about Indian institutions repository, there is also some challenges. The problem is storage and maintenance, and there is also limited coverage, and they are not regularly updated. And also there is no accession system for tracking the resource. So we are addressing this gap with the help of Bioverse. So basically, Bioverse is a unified integrated platform to integrate Indian biological resources at same platform under IBDC ecosystem. It follows FAIR principle, and it also ensures long-term sustainability of resources. It also provides interoperability across bioresources, and we also give national accession to all submitted biological resources. And with this accession system, it gained global visibility and availability. And this is the flow how user can submit their biological resources. So if you have any query about Bioverse, you can write on the support, and you can visit with the site. It is the home page of Bioverse. User can explore it and they will get some insight about Bioverse. And if someone wants to submit their bio resources, the first step would be user registration. So it's hardly 2-minute filling form. User just have to fill basic information, and after filling the registration form, they will get some OTP, and just they need to verify their email, and they are all set to log into the Bioverse. And after login, they will land it to their dashboard, and here they can see their submissions, and they can track their submissions, and also they can submit new bioresources with the tab new submissions, create new submissions. If someone wants to create, there are 4-5 steps. It's hardly take 5 to 10-minute time if they have all the information with them, like title of bioresource, grant number, and publication, what kind of a bioresource it is tool or database, and some categories means in which category it falls, and and we also wants their backend and front-end file so that anyone can download to their system and use that bioresource. After submitting all these steps, user will get IBDC accessions, and here they can preview their resource, and they can also share, and they can also cite their bioresources. So this is the browse and search part. User can go and they can search if they want any tool for their analysis. They can search with this browse page, and they can download the tool and they can use the resource. Here are the future vision of Bioverse. To establish Bioverse as a national platform at Indian Biological Data Center, powered by federated data model for seamless integration of data ownership. We also position India as a global leader in the bioinformatics and biological data science. and to enhance global visibility of Indian bioresources to foster strong international collaborations. We also want to make it as a secure and compliant analysis environment for submitted, manage, and control access data using recommendation through AI for tools and database so that user no need to go some other platform to analyze their data. So this is the Bioverse team. Ravi, he is technical assistant, and Rahul, they are very hardworking, and they completed this Bioverse within 2 months. And I'm the leading lady. I have made this idea and make the metadata. And along with this, IBDC is also active in outreach programs, as you can see in the pictures. And we also have some people here in this conference, they have visited IBDC. And if you can see in this slide, from 2022 to 2026, we have got user distribution across world. And we also organize international and national conferences. Like in 2024, we organize AIBC, and we have got more than 300 delegates there at that time. And computational biology conferences, it is our all annual conferences we organize every year this. And I would like to thank and acknowledge my whole IBDC team, and along with Principal Investigator Professor Arvind K. Sahu, Dr. Devasish Mohanty, Dr. Dinesh Gupta, Professor Deepak Nair, and current head Dr. Sashank Shekhar for believing in my vision and providing all the supports. And I would like to give more acknowledgement to Dr. Rakesh Gokhale, Secretary DBT, and Professor Saurabh Raghuvanshi. He was former lead of IBDC, and he developed IBDC from scratch. And I'm happy that Professor Saurabh is right here with us. And Professor Sudhanshu Vrati, former Executive Director, RCB, and Locus team for HPC support. So if you have any query, any question, or anything, you can visit IBDC and you can write us to the support. And yes, thank you. Thank you so much.
Thank you, Dr. Ayada, for that wonderful presentation. And I commend you for the great work you are doing on Bioverse. So the second presenter in this session is Professor Kyoko Aoki Kinoshita from Soka University, Japan. She's going to present her work on semantic analysis of glycomos, portal integrating glycoscience knowledge, and help me to welcome Professor.
Hello, everyone. Thank you. So yes, I will be talking about something that's not related to genomics, but glycosciences, and I'll give a brief description first. So for those who are not familiar with glycans, these are carbohydrate sugar chains, post-translational modifications that are found all over the cell, every cell. So as you see in the in this slide, we have the cell, and on the proteins and lipids, there are these glycans, these carbohydrate molecules which cover it, and these are used as signals for immunity, for recognition by bacteria, by fungi, cell-cell interactions, etc. And they're known to be involved in various biological functions, including infectious diseases and dementia and tumors. So, I'm missing some slides. Okay, so I had other other pictures about how glycans are synthesized in the cell. So there are many, many different enzymes, glycoenzymes, which help synthesize the glycans inside the from the ER to the Golgi and up to the surface of the cell, and they're attached to not just proteins but also lipids, and they function as modulators of protein function. So it's important to understand the glycans, how they affect the proteins in order to really understand their function. So we've been developing the Glycoscience portal, Glycosmos portal, which includes various sections. It's quite busy, but in general, it's the submissions area and the data resources. And we have some tools and standards, information about tools and standards. In the submissions section, there is the glycan repository, which assigns IDs to glycan structures, called GlyToucan. We also have repositories for mass spec data for called GlycoPost for glycomics and glycoproteomics data, as well as a glycoconjugate repository called GlyComb. But the main area here is the data resources, where we have integrated many resources from around the world based on the semantic web technologies using linked open data to ensure that everything is stored in a standardized manner inside this resource. So we're trying to integrate all these genes, the proteins, lectins, lipids, as well as diseases and organisms that we could find. So the Glycosmos portal, this is also a very busy slide, but the sections on standards is above. We are based everything on standards. The repositories help to populate the data resources, which contain a variety of information. I will talk about this section here, where we have been trying to integrate literature information and populate and expand on the data in the data resources section. So the challenge has been that we have now integrated many open databases, but there's still much information in the literature which need to be mined. It's not available anywhere except in the literature. And so for glycobiologists, they often have to go to the journals and to learn about the latest research. So we've tried to come up with a scheme to be able to annotate automatically data about glycans from the literature and do an automatic pipeline from literature to the data resources in Glycosmos. So as a glance, we have my collaborator at DBCLS has been developing this pub dictionaries resource as well as pub annotation. And then these are translated, the annotations are translated into RDF, which can be queried using Sparkle, and then also visualized and integrated into Glycosmos. And so we have this method of named entity recognition in pub annotation, which can in a smart way also detect abbreviations, such as, for example, RA. If it's used in the first instance, it'll automatically recognize it as rheumatoid arthritis, for example, in the article, and then fuzzy term matching as well. So I hope, okay, sir. So the dictionaries that we prepared for this work as a proof of concept include these 8. So we have glycan motifs, which comes from the GlycoNavi resource as a collaborator of Glycosmos, as well as the glycoepitopes database, and then taxonomy, disease ontologies, phenotype ontologies, and anatomy ontologies as well. For the GlycoNavi, this is the GlycoNavi glycan motif dictionary. It's basically a list of the terms and then the URLs the URLs to link to that information. And what we've done is we also have images, like the graphical images from GlyToucan, which we can show in the annotations that I'll show later. So this helps to visualize the annotations that we obtain. The pub annotation tool can take the dictionaries that we specify and then also specifying the, I don't have it here, the list of publications that we wanted to search through and annotates it using their technology. And as a result, well, results come later, but we have annotated over 650,000 documents which have been annotated with these tools. It's just a snapshot of that. We get the PubMed IDs. The annotations are then all RDFized, and then we have the Sparkle query or endpoint, which can be used to obtain the annotations from within those results. So as a result, we have, like I said, over 650,000 abstracts collected, and 15,000 of them had lichen annotations in them, with a total of almost 300,000 annotation instances and 141,000 sentences. These are the journals that we used, the ones that are most often publishing glycobiology research, and as a result, these are the number of annotations that we could capture among all those publications. So we have a lot of motifs about glycans, which can range from a single monosaccharide, which is the most common, GalNAc, or terms that are found within motifs like chondroitin sulfate, chondroitin, sialic acids, and other often used motifs for glycan structures. Similar for glycoepitopes, and then many taxons and diseases and anatomies. So what we did, this is just showing the disease occurrences. We could confirm that the motifs and diseases that co-occurred within those annotations were already, known to be biologically meaningful. So GM1, a famous glycolipid, is related to cholera, and we know that GM1, that toxin binds to GM1, for example. And influenza is known to use sialic acid on the cell surface to enter and also detach itself from each cell that it infects. And Sialus X is known to be involved in the inflammation through this lectin, etc. So what we could do then from this information is visualize this first. So these are the terms that are highlighted using text IA that can show which terms and annotations were found. It's missing the glycan image, but okay. And so this is then translated into RDF and integrated into the Glycosmos portal. Okay, so what you can do in Glycosmos is we've provided the list of all the articles. We can provide the keyword search, the visualization, which I just showed, and then templates for querying using Sparkle, so that non-experts can just fill in a predefined query, fill in the blanks, and look for sentences showing that some glycan occurs with the disease. We have, for every glycan, we have a detailed entry page in Glycosmos. So now that we've integrated that into each entry page, and we update Glycosmos every 4 months. So this is updated periodically throughout the year. So this is showing the pub annotations page we have in Glycosmos. The latest, this is old, the latest publications, author, PubMed IDs. If you select a title, then the text will show, this is a search using NOE5AC, sialic acid, will give the results and showing the text in which that was found. And then, okay, so the specific entry page, once you select the PubMed ID, it'll display the actual glycan image within the text. So the users who are most familiar with the glycan structures images, they can easily extract that information from the abstract. And then this is the sample of the actual entry page inside Glycosmos for this particular glycan ID, which shows that it was found in these 10 publications. And again, the user wouldn't have to go through the entire list. They can just go to the single entry page and then find that information. So we were able to automatically find a way to extract information from the literature using simple dictionary engineering. We didn't do any machine learning training or anything like that. We could annotate with a smooth pipeline from the publications to RDF and then to the linked open data in Glycosmos. And of course, this is extendable, very easily extendable with new dictionaries. It's all open data, CC by 4.0, with code available under the MIT license. So as a future work, we plan on automating this for other resources, not just glycans, but also glycoproteins for the lectins, etc. And of course, the use of AI agents, MCP servers, will help to fine-tune many of these technologies in the future. So this is coming up. We're trying to get into this as soon as possible. Finally, acknowledgements. Okay, time's up. Okay, so the collaborators for Glycosmos are funding agencies, and these are the resources, but you can ask me anytime for any questions. Thank you.
Thank you. So we just move on because we are a few minutes late, and our next speaker is Dr. Marwa Khediri from the Pasteur Institute in Tunisia, and she's also a member of the organizing committee of this meeting, and she's a member of the African Genomics Data Hub. Okay, welcome. Okay, welcome to your talk.
Hello, everyone. I'm Khediri Marwa, postdoctoral fellow at Pasteur Institute of Tunis. I would like to thank you, the ASB, the International Society of Biocuration, for giving me the opportunity to be here with you for the first time in Cape Town. And today I will present the African Genomic Medicine Portal, AGMP, a web portal dedicated to African ancestry genomic information. It's a project led by Professor Qais Ghadira and Professor Samar Qasim. And our PIs are Professor Qais Ghadira, Professor Yasmina Faqim, and Professor Nicola Mulder. Okay. So beginning with a brief background, African population data are fragmented across multiple biomedical repositories. This leads to challenges in data retrieval, representation, and fundability. So in addition, these resources often lack fine-grained structure and standardized annotation to adequately capture the diversity of African population, which present a clear need for a centralized, harmonized, and well-curated research. In this framework, the AfriGeneD project was established to address the fragmentation and lack of standardization in African genomic data. It stems from variable resources developed through the H3A Bionet project. It aims to centralize, harmonized genomic data platform to cover the full data life cycle from generation to discovery and from analysis to archiving. Also to improve accessibility, interpretability, and fundability, to promote standardized annotation and curation, and finally to support research and healthcare integration. So overall, AfriGeneD project bridge the gap by transforming fragmented data into a unified, accessible resource. Also in the framework of AfriGeneD, the AGMP project aimed to develop and maintain an African-specific genomic medicine portal, integrating and harmonizing data from multiple resources. It aimed to centralize genomic data, to facilitate health research, to support precision medicine, to improve representation, to enhance data accessibility, and also to provide a public resource. In terms of use of cases, AfriGeneD AGMP project support different use cases like researcher conducting drug disease research on African participant, doctor seeking more information on latest discovery and developing science, student early career scientists, senior scientists, policy makers, and industry. So if you want to go in the portal and do the research and see the AGMP functionality, we can do the research by variant, by disease, by drug, and by gene. If by variant, our input will be the RSID and our output will be the gene, drug, and disease affected by variant. If the research in the portal is by disease, our input will be the disease name and our output will be gene and variant associated with the disease. If by gene, the input will be gene name and output will be variant, drug, and disease affected by this gene. And if by drug, the input will be the drug name and the output will be variant and the gene that affect the drug responses. Here I present the AGMP data retrieval and the curation process. So we begin with the raw data collected from different resources, essentially from PharmGKB and DisGenet research. Then we collect the PIMD from different publications from PubMed. And after that, we do a primary filtering for geographical origin using text mining method. Then with a manual curation of PubMed abstract, it's necessary to extract key information like geographical origin and p-value. And in the same time, data wrangling for gene, protein, and drug data annotation are extracted from different research like Ensemble, Uniprot, PubMed, DrugBank, and SHMBL. All this curated and harmonized information are integrated then in the database of the portal of AGMP. Unfortunately, the data was collected through a combination of automated text mining and manual curation. Concerning the text mining pipeline, to exclude non-African data from GMP, a custom text mining pipeline that identifies biomedical literature containing African-related biogeographical and ethno-linguistic terms from midline abstract was developed and applied to the retrieval dataset. The pipeline used the adirect tool to retrieve the PubMed entry in XML format, followed by a series of processes to tokenize and limitize the text content. Finally, the list of biogeographical and ethno-linguistic terms was queried, and the abstract that matched at least one of the terms were retained for manual curation, and the pipeline is implemented in the next flow workflow manager. Concerning the manual curation steps, it was included in the content curation process due to false positive, which may have resulted from the text mining pipeline. This step was also employed to harmonize information related to the biogeographical group of studied and to extract information about the country of origin of each study. Cohorts of mixed African origin were assigned to sub-Saharan Africa or Africa depending on the geographical spread of the study sample. Study p-value were retrieved either from the PharmGKB metadata field or by recovering it from the main text of the paper. Concerning the AGMP data integration, it consists of applying the extract, transform, and label procedure to integrate data from different sources into a relational database. A series of interactive transformation code were applied for this purpose. Manually created data were examined for their consistency of using unique terminology in each column. All the data found to describe a population from non-African ancestry were discarded from the source table. Here I present the quality assessment process provided by the AGMP portal. We begin by evaluating the fraction of missing data. Then we process in harmonization of terminology. After that, we do also harmonization of class label and the presence of duplicates. And finally, we verify the correctness of mapping to the target tables. Here are present the different the genetic data entry in the portal. We have until yesterday more than 2,000 genetic data collected from two sources, PharmGKB and DisGenet. Also, here are presented data overview of the information collected in the AGMP portal. We found a total number of 6,270 genes, 48 drugs, 1,579 phenotypes, 70,470 variants, which are collected from 1,194 unique public and unique studies. Concerning the latest functionality added, we have added the mapping ethnicity and geographical information to AFPO, the African Population Ontology, for more refined African data searching and exploration. Also, batch querying the portal, we support this. Then the metadata submission from the African research community and the investigating LLM enhanced data mining pipeline to retrieve publication not captured in our existing data resource. Here are presented, you can find in the portal the top 10 drug, the top 10 gene, the top 10 variant, and the top 10 phenotype in different metrics that you can find it in the portal. Also, concerning the ethnic diversity across study, a total of over 90 distinct ethnic groups were identified across the study, highlighting the population diversity represented in the dataset. And here are present the metric by Google Metrics, where we can find the user geographical detail. For example, here, the top 5 user geographical detail are from South Africa, from China, from Singapore, from United States, and from United Kingdom. Concerning the GMP ongoing work and the next step curation, we plan to investigate how to incorporate African-specific data from CleanVar PG catalog, how to integrate data from the Agora TM project. We plan to add the TCGA database. It will be useful for such study. Look also for new published African data not figuring in the existing database, and to develop the procedure of data curation to follow. In the other side, we plan to update the PDF tutorial, create video tutorial, and update the documentation. Here I present to you the portal, the African Genomic Medicine portal, the link to the interface of the portal and the link to go it. You can do your search by variant, by gene, by drug, by phenotype, and you can explore the curated data by this link. At final, I would to acknowledge our project lead and our project manager and our PI and advisor, also our contributor, and thank you for your attention.
Again, we have time for questions after the next and last session. Talk by Lynn Schwimmel, who is an associate professor, full professor at the University of Maryland Medical School, and she will talk about human disease ontologies. Thank you.
Great. For over 20 years, we've been building semantic infrastructure for data exploration of human disease knowledge. Today, I'm going to talk to you about and share with you how we've been modeling and integrating environmental drivers of human disease into this system. Much of this work is driven by our user community, their needs and their interests. Here I'm showing you a recent analysis I put together from our user base. For many years, we have been mining and curating who cites us in publications and who uses us, and that information is available on our website. For this talk, I wanted to think about it a little differently. So taking all of those publications, creating a data set, running it through ChatGPT, I want to see what ChatGPT could do to identify large-scale themes of the work. Because we curate the themes, but we have probably 20 categories we would humanly curate them to. So utilizing ChatGPT, I found it quite interesting to see how they broke it down over time. And then when I turned that into an Excel graph, I was like, that's really interesting. It does really represent our earliest days of representing the data as an ontology, the lower blue. And then as we've continued to do that, that's great on the bottom, but as you go up to the top of the slide, you'll see some of the newer user areas that weren't really present at the beginning and are really driving the work today, AI and machine learning, but pertinent to today's talk, the top line there, the multi-omics and the biomarker discovery. So it's really been interesting to see how we've broken it down to 3 eras, and this is really just taking the dates of the publications and their titles and seeing how it kind of came together. So informative for us for our planning, but interesting in and of itself. This is our homepage. We revamped it this last year. You can do searches right from the homepage, but I'm pointing this one out as a navigation if you want to come check out our site. The trees are available at the top under disease ontology, and the tools I'll talk about are under that DOKB or disease ontology knowledge base. Our system has evolved quite a bit over the years, from the left to the right at the bottom of the slide if you take a look. We initially started with the mandate of putting a classification system together of all human diseases. Putting this together by the cause of the disease and trying to understand what was the most appropriate home within that classification for every disease. As we progressed across the slide and across time, we wanted to further that classification by understanding what were the cells of origin of some cancers, what were the anatomy sites for every disease that we could find, and so on and so on. I'll show you there's quite a long list of these other types of mechanisms or features of disease. We want to improve our representation of each disease and its various sets of information, but how they're related to each other as well. That helped us transition to a knowledge graph at the far right of the slide, where we've integrated this graph structure into how we represent the data back out to the world as well, but allowing that semantic richness to be queried and mined, as you saw on the last slide, quite a lot. But the idea is that you should then get into the ontology itself, where that is our data model, which we put out with our monthly releases, and there's stats on the bottom of the slide. We continue to add data every week. We get requests through our GitHub repository all the time, and then we integrate this information for the world to see. Here I'm showing you the different versions of the ontology, because this is really where we put the data in, and this being a curation meeting, I want to show you how we're curating the data, what our thinking was. So we have our ontology etiology-based tree on the left, in the middle, all the different imports of other types of data that we then annotate onto those diseases to be able to represent how they're related to each other. And on the far right, I'm showing you our curation kind of workflow. It's a framework of how we envision bringing in new types of data. Walking through it, we do a lot of review. We reach out to other teams and clinical communities to make sure we're representing the biology correct. Then we integrate it into the ontology mapping. And then further, we do the annotations within the ontology, and then we have these graphical representations I showed you before, and I'll show you in a minute. And then we have AI-ready data sets. This is something I'll show you that you can pull out of the system through programmatic queries, but it's also part of our monthly reporting as our data release. We have in our GitHub, we have a folder called DO reports. So you can just get these data files if that's easier. We also do bespoke ones upon request. I want to go through a little bit of the modeling of why we were doing these environmental drivers and how we were thinking of them. So this is work done by my clinician team, and I'll show you 3 slides from that publication here. We really wanted to look at the spectrum of diseases, from ones that are purely genetic in origin of their cause and those that are purely environmental and all the in between. So we want to make sure we were modeling the full spectrum so we could really understand what are the elements, the data elements we need to pull in, and so we could understand them and represent them appropriately. This is one of the tables from that paper where we're really starting to look at identifying examples of that full spectrum, from ricin poisoning, fully environmental, Tay-Sachs disease, 2 we know right now at least, fully genetic, and in between. This allowed us to start to put together a data set of hundreds at this point, starting in early hundreds, of diseases where we knew the cause, an environmental cause directly, not an association, but an actual cause. We want to be quite rigorous in what we're putting in because the level of knowledge there's so many environmental exposures out there, right? We want to be very careful what we were representing within the database for when they were known to be causal between the two. And then again, we had to reach out to further in our clinical community to do this work. This figure was put together for that paper when we were starting to think of all the variety of environmental drivers that we may want to capture so that we could start thinking about how we're going to put them in the ontology. Where could we also get the support within literature to represent that data? And so this is an ongoing progress of work we've been doing. The far right of the slide is showing you a very small little ontology put together for this work to bin the types of environmental drivers so that we could categorize them and get our heads around them, how they differed. So again, will allow further data exploration. And then we're using quite a variety of other ontologies because the data we found about environmental drivers is been characterized in many different systems. So we're continue to bring those in and annotate them onto the diseases. I did want to show you one of our graphical views. For every disease within our system, we have this representation. You can play with the graph view, or some people quite like to play with that visually. Here I'm showing you the full tree on the upper left and the different parts of it. And then I'm showing you the disease driver ontology pulled out into the chemicals and the biological drivers. Again, this is just giving us a framework to capture that information, and it will be evolving because the knowledge will be evolving. So keep an eye on it. It'll continue to grow. I want to show you a few of our data tools where you can programmatically mine this information, depending on your comfort level, the types of ways you like to do analysis. We did our API this last year, so it's quite rigorous now. There's a lot of variety of what you can pull out using the API. I'll show you the Sparkle sandbox that we have to be able to play programmatically with that data, and then a faceted search, which plays with the ontology information. Again, all providing different ways to pull that information out, ask different questions in a programmatic way. I also find it quite useful for the curation as well, to have these tools available and have specific curation questions I want to look at. Okay, the Sparkle sandbox briefly. We have a number and growing number of Sparkle queries that have been vetted and already available for you on this site. And when we have a user ask us for something new, we add it to the site as well. And these are available in our GitHub. What this allows you to do is to pick one of the pre-canned queries, and then you can actually edit it. I find myself doing that when I'm doing curation. Maybe I have a query here on synonyms, but I want to do some other data element that we have. And you can just edit the query. It's all in there. And then it produces your AI-ready data sets because every element within that data set is rigorously attached to another ontology or the disease ontology. One other thing I wanted to mention on here, as you can see on the far right, the box I have in orange, we can also do federated queries. This means we have devised linkages to other resources like Celosaurus, like Wikipathways, that have Sparkle endpoints like we do, and then you can query both our data at the same time if you're looking for data sets. And our faceted search, this is an in-house tool we built. We wanted to have the ability to walk up that ontology tree, because anyone who's worked in ontologies, you know, OWL is kind of an esoteric kind of language to deal with. It's not necessarily a file you can just easily look up things. So we want to be able to have a way to go up the tree and capture all of the annotations along the tree, because children inherit the parents' annotations or features in an ontology. So in this case, you can select any of the features, the numbers there indicating how many annotations there are within the ontology for that. In this case, I'm showing you disease drivers and OMIM susceptibilities, which are our genetic drivers ontology. And then looking at the set of diseases that have that overlap between the two, or that intersection. So it gives you another way to look at the information and pull out data sets that you're interested in. And this, lastly, ending with my team. I mentioned our clinical team. There are others involved as well when we have queries or interest in a certain area. And then my it just turned off. Okay, you saw them all. The informatics team on the bottom. These slides are on Figshare already, and if you want to join our public Slack, there's a link in our various social media, and there's a QR code too. So thank you for your time.
All right, I guess we have 10 minutes for questions, and maybe all the speakers can up to the podium. Needs help?
Can we just take it down, bring it down? Would that be easier? Yeah. No, we just do it right here. Yeah. Sure. Oh. There you go.
Okay, any questions?
one.
Hello. Thank you so much for all of your presentations. And oh, sorry, I have to look up the name. Sorry, Dr. Yadav. Thank you so much for the presentation on Bioverse. I'm very excited to see the resource you have put together. I wonder how, if there are any plans, and you had a slide where you presented all of these gaps in so many of the other resources. And so I wonder what's your vision to perhaps feed back information to all of those resources, or how do you plan to let the community know how you have filled those gaps?
Yes, we have got some feedbacks, and on the basis of those feedback, we considered the gaps and created filled that gap through Bioverse. I think you asked this question.
Yes, and what is your best way to you have provided the feedback back to those resources, right? And so they
Yes.
know. Okay, thank you. That was it. Thanks.
So thanks for all the talks. I have a question for Professor Aoki Kinoshita. Here. So my question is about the co-occurrences of glycans and diseases in papers. Can you elaborate on how you define them and how you weigh the evidence?
That'll be for a question from a collaborator, but I believe it's the distance between the terms in the publications. So there's a parameter for that to determine if two words are co-occurring or not.
Okay.
Yeah.
Thank you.
My question is for Lynn. As an external user, sometimes I'm confused on when to use DO versus HPO. And I wondered if you could maybe shed light on when you see which ontology is appropriate, or if we should use them in conjunction.
I would say in conjunction. They have different points of view and purposes. HPO is human phenotypes. And then in the clinical setting, phenotypes are features of a disease. So when I'm looking to describe a disease, I'll use the HPO terms to describe the various clinical features that I see.
Okay, thank you. So my question is for the AfriGen project. I was wondering, are you precisely dealing with the gathering or the defragmentation of real data or just metadata? And if you are dealing with the gathering of real data, how are you dealing with, I would say, the diversity and the fragmentation of regulatory policies when it comes to the sharing of data from one country to another country, precisely in the African continent? Thank you.
It's metadata. I believe that we introduce the data from different resources, from GKB, DisUnit, also from the GWAS catalog resource, and it's really metadata, and that's all. I can invite Dr. Qais Ghadira, it's the PI of our project, to respond to you if you want.
I'm one of the PIs, so with Nikki and also my colleague Yasmina. So we have a huge team working on that. So it's just, we are not dealing with data. You mean raw data or genomic raw data? No, we are not dealing with that. So we are collecting data from papers or also from several databases like DigiNet, PharmGKB, and actually GWAS catalog. And we are curating African related data, which are very, very hard to find. So yeah, we are doing this, and we are injecting this in our portal that you can find easily on the African Genomic Medicine portal. Yeah.
So thank you for all speakers for your insightful presentation. My question is for Dr. Marwa Khadiri. What is the functionality or features that you plan to add in AGMP portal?
Now we are working on drug, disease. Also, we are working on the drug, on the disease, on the gene, and we plan to add many others features, but we didn't precise them. We will discuss this and we will add them. Yes.
You said that you plan to add a PO and
It's from the African Population
Yeah.
Ontology portal.
Hi there. My question is for Lynn. So me again, hi. So really cool what you made starting from an ontology, then going to a knowledge base and like from like identifying the causative agents are mental or genetic or whatever. But you can do so much more in theory. So what's your starting point? What's next? What's coming up?
I don't want to give it all away. We're ending our renewal this year. So we keep an eye on where the science is moving, and we mine those publications and the groups that are using us, and that really does inform. We have quite a laundry list right now of what we're going to move forward on. A lot of it is making more data connectivity, and then I'm going to stop at that, but just because, anyway, it'll go quite long. But we do continue to see where we need to move the data forward to make sure the analysis can be done by all the groups.
Cool, thank you.
Yeah. One thing I could say is that we continue to make sure we're representing rare diseases around different communities. So that has been one of our pushes the last couple of years.
If I can ask another question about BioWorth. So I imagine people upload flat files, right? So how searchable is that database? So if you have heard of biostudies from the EBI, I imagine it's similar to that, right? So it's not a relational database, right?
Bioworks basically to submit tools and biological tools and databases. So all the codes, means front-end codes and back-end codes, we submit in the Bioverse so that user can download those files and they can install that tool on their own system and they can run for their analysis.
But they can download individual data files, right? They can't search across BioWorth, across
They
different
can search,
data.
they can search also. Suppose someone wants to analyze some kind of data. So they can go on Bioverse, they can find certain type of tools and databases. From there, they can download those tools and analyze.
Okay, thank you.
Yeah.
Questions, I thank the speakers again. And if you want to, you can, of course, pester them during the coffee break. Thank you.
Excuse. There is another speaker. Okay. Please don't go away. We have one final speaker, a few minutes from Inkaba Biotech, a representative from Inkaba Biotech. Shakira Lucas, you are most welcome to give us a talk.
Okay, hello everyone. Thanks for staying. We have reached the end of our scientific bit for the day. So you can go back to your grants and manuscript edits if you want to. Anyway, my name is Hamilton, and for the detectives among us, you will notice I am not a Shaquila. Congratulations. My name is Hamilton, and I'm just standing in for Shaquila today. So I just want to give you a little story about the company that I work for. We're one of the sponsors of today's event. So we started in 2002. We started with the oligosynthesis services, and we had 2 polygen synthesizers, and that used to have a capacity of 10 oligos per run. So we didn't do much every day, but we started small. And for the seasoned, I don't want to say old, but the mature scientists among us might find that other word, spectromedics, familiar. But that was, I think, one of the early or pre-Sanger days of sequencing instruments. Anyway, we had one of those too. So we did oligos and Sanger sequencing, and we serviced the immediate Pretoria region, and we tried to fan out to other parts of the province. And then a very important detail, at that time in the company, if it was my birthday, I could buy a single cake and leave it in the kitchen in the morning, come back in the afternoon, and have a few pieces left over to take home. That's how it started. Then in 2007, we acquired the first NGS instrument on the continent, I think. If I'm wrong, please tell me, but I hope I'm right, because this is a good sales pitch to know that we are the first ones to start NGS on the continent in a private capacity. Anyone know what that instrument is? The CBD points for this. Okay. 454, which one? It actually was just the titanium, actually. Yeah, correct? Okay, good. So that's where we started off. We used to produce like something like 20 megabases or something pretty impressive for 2007. And then we decided, you know what, as a company that provided services, we actually need to sell stuff as well, because we cannot just sustain ourselves on services, because NGS was in its infancy. And so we started to sell stuff, reagents. And anyone know what this company is? Anyone find this company familiar? No, but even stranger, anyone heard of the company, the country, Lithuania? Big country, small country? It's actually, if you spin the globe, you'll definitely miss that country. It's a tiny country. I didn't know it existed until we started distributing for them. Then Thermo Fisher, Thermo actually bought that company, and now all their reagents and technology are under Thermo. But that's how we started off. We had a single product under our portfolio, 2007. And it's the understatement of the year, we have been growing. As of today, this is our life science consumable portfolio. I don't have to introduce these brands to you, but these are some of the brands we distribute under that umbrella. NEB, you guys know. Mirus, I'm sure you know. I'm not going to talk through everyone. In terms of instruments and software, I don't have to introduce these companies to you as well, but this falls under some of our instrument and software portfolio. One of the big ones here is PacBio. We are from 2018, we were the sub-Saharan sole distributors of PacBio. And there's some more news on that in a bit. But Eppendorf, a bit of an obscure company, but no, I'm kidding. But yeah, we actually the distributors for Eppendorf in sub-Saharan Africa as well. And we branched off into molecular diagnostics. No introduction needed for these companies, but you get the point, right? And in 2020, we celebrated our 20-year anniversary. I want to give ourselves a clap, but I'm very, very excited to be part of this journey. As part of that 20-year celebration, the management decided to bring the whole company here to Cape Town, and we had a ball, and it was a real achievement for a biotech company, a biotech startup, to survive that long. And wow. And the story hasn't ended. So, Pulse. We now cover over 30 countries. We actually cover the entire African continent. There's some of our offices. We have 2 in South Africa, 1 in Cape Town down the road here, and our head office is in Pretoria. And those are the countries that we cover, and over 200 staff as of today. I'm not sure of the count because, well, I don't even know all the people, but and we actually distribute for over 120 companies. I don't even believe it myself, but we have multiple people taking care of multiple portfolios. That's how we're able to cope. And then that SKU, anyone know what SKU stand for? Aiton, well done. You don't have to believe me, but you can. I asked our stock controller for this number. We, in our catalog, have over 497,000 units that we can actually quote on. That's how huge our portfolio is. But you're not going to buy every single thing. Some of these are just little screws that go under a centrifuge, so it's not. But even at half that number, very impressive, right? And needless to say, one cake doesn't suffice these days. When we do have cake, we have to have a little truck ship in. And we have hordes of people coming in with the cake, and it's amazing to see this growth. And one of the drivers, one of the motivations for us as a company, one of the goals is for us to be a one-stop genomics shop. And I think we've really come a long way in realizing that this is some of the faces that work for us. And if you look on the map, you'll see that we are peppered across the continent. But you notice there's something missing on the top green. That's Tunisia and Morocco. I know we have people from here, from there. And as of a few weeks ago, we now have become the distributors for PacBio for the rest of Africa. So now we handle North Africa as well. So basically we have entire Africa under our portfolio, and happy to work with our new friends from these new countries. In terms of business, these are the main categories under which we function. And I just want to look, there's a lot here. You don't need to read all this, but under manufacturing, the last point there, very unassuming, but very important, plastics. We actually, as a company, have currently embarked on a massive endeavor to produce our own plastics locally. So, well, I don't deal with that stuff, but it's your 1 mil and your 200 microliter and your 20 microliter plastics and your trays. I don't even know all the stuff, but we are going to actually do this locally. And that's good stuff, right? Because that means a price benefit for our customers and for us. And so we can produce genomics data and run services at a reduced cost. So it's good. It's a benefit that will filter down to everyone, I'm sure. Look, I don't need to read this, but I just want to say, coming to this conference, I actually didn't know what, I'm going to betray my ignorance. I didn't know what biocuration actually meant, to be honest. I thought, like, okay, you go into a museum and make sure you write the right name under that sample or that insect, and before you put it into the drawer. But after hearing the talks and hearing the number of databases that's represented in this room, I feel like nothing less than a leech. Because I realized that in my scientific career, that without the work that you guys have done in creating these databases, like the Go, and by the way, Prof. Lin, okay, this small rabbit trail. Anyone heard of the disease called Stargardt's disease? I'm so glad, because it would be bad if you didn't, but it's anyone else? My neighbor actually a month ago, my neighbor's daughter, 10 years old, was diagnosed with Stargardt's disease. And she, in less than 10 years from now, will be completely blind. How did they know this? They couldn't unless these databases had that information. And so she did some genetic tests, and the ophthalmologist and optometrist, they went to these databases to look for the information. The clinical geneticists also leverage the information in these databases. You guys are doing an amazing work. I think that's why I said the last line, thank you. Because I said I feel like a leech, right? You know how much data I worked with, but all of it would be utterly useless if we didn't have these databases. Thank you so much for the work that you do. You know what? It's for me, a big wonder is why is this particular conference so small? If the government knew how important this work is, well, we should have a bigger community here. But I just really want to extend a thank you to you from Incaba, from myself as a student. Without your information, the data that I worked on would be utterly useless, sitting on a hard drive somewhere and having no value to anywhere. So thank you to you, and thank you for listening. You're released to have tea if you want, but that's me. Yeah, thank you.
Can I have my slide up? It's such a great endeavor to have a one-stop shop for genomic. Some of my collaborators in African continent, they complain about the supply chain. You order something, you might take several weeks or even months to arrive. And by the time it arrives, the shelf life is already shortened. So how do you overcome this challenge? And also, how do you, is it your company also arrange courier to enter the country to deliver the consumables, because at the custom could be quite a lot of hoops to jump through.
Anyone got that? So your name is? Stephanie's wondering if the reagents are going to come all thawed to you after you audit. Time is important. Yeah, shelf life is important. No, of course, those are crucial aspects. And I have to say that we've worked hard, and we have established cold chains in places reaching across our current complement of countries that we deal with. And you know, recently we've actually introduced this thing, or this I don't know the name for it, but essentially, when you get your stuff from us, you have to fill in like an evaluation form to say, good, not good. And overwhelmingly, the response is that, thank you, the service was fast, reagents have arrived in the condition it's supposed to, and we could prove and track the cold chain, the transport. And so, good question, and it's always something to be aware of. And we know for ourselves, because if we use reagents that are at room temperature, which is supposed to be at 0, we've got a problem. That's money gone down the drain. That's time. So we make sure that you receive it the way you should. So thank you for the question. Yeah. I was going to say something I forgot because you already clapped for me. But yeah, I just want to say we actually there's something you didn't know, Prof. Lin. No, you don't know it because it's new to me as well. Like for 2 months ago, we actually got customers from Stanford University inquiring about our PacBio sequencing. They wanted to run their services on our instrument. Apparently, the Americans love the RAND more than the South Africans. No guesses why. And so we are certified, so that means the quality of the data you get from us is just as good as anywhere else, because we are trained and certified by PacBio headquarters themselves. And you guys all know the PacBio review. Look, we all know that the Illumina sequencing is very cheap, but I don't know if you know this, but the price difference between long read and short reads are actually similar. now. So many people are actually opting to do hi-fi sequencing. Obviously, the Revio instrument is the high-end, the flagship instrument, but I don't know if you know this already, but PacBio has released, it's just over a year ago, a desktop instrument called the Vega, and you can run, it's made more accessible, but basically all the technology, all the assays, all the applications that you would run on the Revio can now be run on the smaller instrument, lower threshold to enter the market of high fidelity long read sequencing. So, oh, that's some other stuff we do. I won't get into that, but we don't just do sequencing. And thanks to you. That's it.
Okay, are there any other questions? I would recommend to just pester Hamilton at the break, and we can have a break and coffee at the same time and ask questions also. Okay. Thank you.
Yes, just also to mention, we'll have tea break until 4:30. And just a reminder about the bus tonight to the conference dinner. It will leave from the reception. Please meet us at the reception hall at 6:30, but try to be there a bit early. Maybe 6:15. The bus will leave at 6:30 to the conference venue, dinner venue. Thank you.
Sorry? I can't hear you. Yeah, I will put it in the folder. Can I use this one? Okay. I don't.
I think you're doing this on purpose. Every time I come on stage, the microphone right here. Hi, everybody. We are going to continue with the seventh session about databases, knowledge bases, and knowledge graph. I'm Victoria Nunes, your co-chair, your chair, actually. We are going to be hearing from Mr. Aiton Meintjes from the University of Cape Town, South Africa, and he's going to be telling us about HGVD curation, quality control, and annotation of African genomic variation datasets.
Okay, thank you. Works as okay. So thanks everyone. It's quite intimidating looking at the following talks. So I'm going to be just talking more about African, as the title says, and explaining some of the frustrations and some of the things we have adopted to address some of them. So AGVD. It's meant to be an African nomad. So it's driven by the fact that the global variation frequency data use human variation. The global variation data sets often don't have a lot of African samples, and so they often all get lumped together as just African. Whereas there are more samples available. And so what we've done is sort of focused on that and tried to serve those in more meaningful ways for people working with African projects and cohorts. So that's just an example of, for instance, Nomad, where it's just listed as the frequencies are African. Whereas there's, as we know, there's a lot of variation in the African populations, and there's some nice clustering. So we're hoping to be able to leverage that by putting a bit more attention to the samples and how we report the frequencies. Okay, so that's the use case why we need this. AGVD, we have the interface, we are serving up frequencies. That's not what I'm going to talk about today. What I want to talk about today is about the actual curation part. So within AGVD, we have three types of data that needs to be processed and sort of used. So there's what we call a joint call set. That is roughly at the moment, it's 4,500 samples, high coverage from very many different projects that are joint called together and give us a joint call set of, I think it's in the order of 140 million variants. That is what we report our frequencies on primarily, but then we also do have, and we know of, studies, additional studies that have been done with genotyping arrays and exomes, so on, which we could leverage and add. But we're keeping in mind that the nature of those frequencies is, the quality, not the quality, but the nature. So we can't just lump them all together. We don't want to lump them all together. So we have these two paths of genotypes coming in, and then finally the annotations, which mostly we fetch all the annotations, but we do have some of the annotations on created by ourselves. So I don't know if people can see this, but just to give you an idea of what we start with and how I was trying to make this neat. On the left-hand side is just a screenshot of the different projects that are contributing. So quite a few of these projects are publicly open data, so 1,000 genomes and so on. But it's just to give you a sort of an idea of when you get metadata with these from these different, even though it's all sequencing projects, the nature of the metadata is different for all of them. So I've tried to sort of regularize it and then systematically import. On the right-hand side, you can just see the types of properties that the different sample metadata files have. So I'll get to that a little bit sooner. So what I'm talking about now is the post calling. So it's after the joint call set has been produced, and that's actually upstream by a colleague of ours. So this is sort of, you know, that's how it's calling, and we've got this big square joint call set. So a bit of work to collate the projects, what I was showing you there previously. Something I do is I mask the sample IDs. I think that's good practice. Then we start to build up a metadata table of, it's not a lot of metadata, but it's what we have. And then finally, we go through that list of samples and make sure that there aren't some stragglers or problematic samples. So, for instance, out of the 4,500 last time, I did remove 4 samples. They weren't our samples, they were actually HTTP samples due to ID problems. And then we do a little bit of a minor amount of variant filtering, because obviously the filtering's already been done. And actually, the call set comes in with annotations that have been added, but we've decided we strip those out and rather systematically run annotations again ourselves. And then we do frequency calculations. And some of the tools we use, Jupyter extensively, but also we're quite familiar with Nextflow. So many of our workflows, not this one, but many of our workflows are in Nextflow, and we're looking to add Nextflow to it. And then from our discussions with Nomad, which is much larger projects, they've moved on to a distributed table processing system called Hail. So we're anticipating that we're going to need to do that also as we grow. Just an idea, this is all from the same file, but just to give you an idea of this is what is the regularized metadata that's coming in. I've put the sort of anonymized the sample IDs, and then there's ethnolinguistic group. But I just wanted to point out the differences in detail. So 1,000 genomes is very well known that, you know, that population is English and Scottish. Another project of ours, or a collaborator of ours, just mentions colored or black or white South Africans. And then another project where the sample is smaller, this seems to be very sort of low, detailed population group information. So that's a challenge and something we carry, well, we try to address, but it's an interesting problem. Okay, so that's the joint call set. I have 3 minutes. That's the joint call set, the first type of data. The second challenge we have is other projects coming with chip data sets and wanting to contribute it. And those are very valuable because they often have many more samples. But the problem is each genotyping array set, it's a project in its own to do the calling. You need to look at the data itself. So at the moment, we've adopted a policy of we want to receive the called VCFs, and then do a little bit of QC ourselves, but we don't want to have to go through the whole calling process because it is nuanced and it takes time, and we're going to end up with different calls from what the published data set has. And then finally, the annotations. So we add annotations on the variant level. So each variant, we add annotations, which is the frequencies we have calculated. So I can't remember if it's in the GUI, but like for instance, we've clustered the population. So we've got Central Africa, Eastern Africa, Northern Africa, Southern Africa, and Western. So that's already a little bit more detail than is often available on the international sets. So pre-calculated allele frequencies, we are adding, we show that, and we've got some other ideas or ways of looking at it. So instead of this very rigid east, northeast, southwest, we're also looking at PCA and clustering to perhaps make more informative assignments of samples. Some of okay, so the bulk of our annotations we actually fetch from a service called Cellbase, which actually checks a lot of annotation databases. So we do that, and then some extra data sources like DBSlip we fetch ourselves. And we're probably going to move over to fetching more of the different big data sets ourselves, the annotations. Challenges, so like I said, the disparate data sets, even in that joint call set, it is samples from lots of different projects and different types of projects, different sampling strategies. So you've got 1,000 genomes where it's very nice and square. There's 30 of each group. And you have this sort of more diverse human genome diversity project where there's 1 or 2 from some groups and so on. Data size is not a challenge yet, but we definitely, I mean, if we're looking at 4,500, when we get to tens of thousands, there's probably going to be some more optimization we need to do. Versioning, it's an open question. We haven't decided how to do it, but ideally, if somebody goes to AGBD 2 years ago and looks up something, if they come this year and if they're publishing, they might want to refer back to that old version. So we're not too sure how much we need to preserve of that. And then we're definitely seeing a growing need for being able to custom slice the data set. So in other words, we've got this big joint call set. We chop it up into 5 and my time's up. But I'm done. Yeah. So that's what the hell is for. So thanks.
Thank you so much. Now we are going to hear from Dr. Sushma Naitani, who is going to be telling us about Plant Reactome Knowledge Base 2026 updates and future directions. Thank you.
Hello, everyone. I'd like to thank organizers and all of you who stayed for this last session. I very much wanted to interact with you, but I have a scratchy throat and I was trying to keep away not to infect anybody. I feel better today. Here I have 10 minutes and I'm going to represent a couple of things here. So plant reactome is one of it. And before I go to plant reactome, I like to acknowledge travel fellowship and support that I received from OSU President Commission for Women and also from the Ag Biodata. So together, these 2 fellowships made my travel here possible. And I like to also acknowledge our partnership with Grameen Database, with the Human Reactome Project, EBI, and more recently, I have become also editor for Genetic Society of America's journal Genetics and G3. So anybody, one of the things is that we have a computational biology section and we are really friendly. You can submit your papers in any format and we are very quick. So if we have to refuse, we can refuse within a week and we are very friendly. So hopefully if you have any papers and considering for genetics or G3, you can contact me. So again, I serve as a steering committee member for Ag Biodata Consortium. It is a consortium of about 44 biological databases that are related to agriculture. And what we do here together is that we look at the data, we look at the resources, and we see where are the things that we need to work together. So we don't duplicate things and we don't struggle alone. So it is a partnership that we can look into the data, standards, metadata, formatting, tools that can be reused and making data fair. So this group started in 2016 and officially we have a RCN grant from NSF. And it was quite a pleasure working with this group. And we have published about 5 or 6 white papers on different aspects. And we have the working groups. And for example, I was one of the working group member for genotype phenotype data. And so there is a paper on the right-hand side that one can find. So now I come back to plant reactome. And so we have many different data types being generated with the advent of omics technology, rapid evolution of the sequencing technology, genotyping, and so on. And these are discrete data. We have many archives that archive these data, these primary data and raw data. However, right now we have only two broader framework or conceptual framework to integrate this heterogeneous data. And so genome browser is one of it, as we know, and then the pathway data is another one. So we are secondary knowledge bases, and we try to absorb as much as different type of heterogeneous data and try to make sense of it and map it to and relate it to. So a lot of knowledge synthesis happens in plant reactome as well as in the gramine that serves as a 223 genomes of plants. So here is the plant reactome, and we work pretty much, we are a public database, and we exist because we are a public database and we collaborate with other public databases. So we curate in rice as a reference species, and then we use gramine and plant ensembles compara tools to do the orthologous mapping to other species and project those pathways. So similar concept that is used by Reactome, and we use the tools developed by Reactome Human Reactome Project. We use their central database so that some of the components that are there don't need the duplication, and our contribution can remain also in the Reactome central database. We connect for the proteins to the UniProt. We connect to the CABI for the chemicals and metabolite and PubMed. And for the plant ontology, we connect to the Plantium project. We use also real-time data from the Expression Atlas, EVI Expression Atlas, and bring it here. So overall, we try not to duplicate efforts, but to connect to other public databases, and then whatever curation we do, it is an added value that we share with everybody. So we are fair and we are free, and we provide access through the API. All right, so this is the plant reactome homepage. We have 139 plant species for which pathway data is available. Users can upload their omics data of any kind, do pathway enrichment analysis. They can compare the pathways and reactions across the multiple species and gain some evolutionary insights. And then we have documentation. We have also video tutorials, and we are able to help you. There is a contact form there, but one can also email me directly, and we have a couple of videos and things like that. So typically, I think many people are familiar here with the reactome. We use a similar Neo4j structure, and we have the pathways and reactions that are organized using the ontological concepts. And this is the upper level firework diagram. Initially, we started with the metabolic pathways, but as we grow, we are using more plant developmental process and curating them using the reactome model. We are also using biotic abiotic response, response networks, transcriptional networks, and so on. So typically, if you open this, we go to Oryza sativa by default, but then one can also select the species of your choice. And this is a typical view of the Reactome pathway browser. We try to curate things within the architecture of eukaryotic cells. So we say exactly in which location, what reactions is happening, how different metabolites or proteins are being transported across the different locations, and so on. And on the bottom panel, we provide the description and summary and citations, molecules, details, and we connect to the CABI and then structure and expression and all. So one can browse. I will not go into that detail here because of the time. But this is just example of the projection of the basal level gene expression data on the pathway. So one can select any of the protein that is depicted here and can see the gene expression data that is we are pulling from the EBI. There are other ways one can analyze data by uploading metabolomic or transcriptomic data and do pathway enrichment analysis. And here, especially in the plant, it is very good because plants have large gene families and not all family members are expressed at the same tissue during the same developmental stage, but there are multiple. So this tool allows us to pick the candidate genes for a particular trait or particular response. There is another way. So we understand that the curation is a very slow process. And so we try to bring the gene-gene interaction data, and we can provide the, and we can also connect this data from BAR and other resources, but users can also upload their interactome data and can find additional connections to the pathway or genes they are interested in. Another way one can look into reference pathways and how they are being projected to other species, and look into the number of the orthologs, if they are similar or if there is a difference in number of the ortholog mappings across all species. So one-to-one comparison, but then one can also download data and look at the whole evolutionary perspective around 139 species and see which reactions are conserved, where we see polyploidy and where the things are missing. So now a little bit about the updates. So we are now looking into updates in plant reactome, and we have updated our biocuration approach. And so one of the things that you will find in the next release is that from the current rice reference knowledge graph, we are going to curate in 15 species for which we have plenty of publications and omics data available, and going to move as a Vidi Planty reference, and those will be projected. So this will give us a lot of flexibility to capture the pathways that are missing in rice, and especially, for example, you have a lot of pathways in tomatoes and fruits and so on. So these are the 15 species. And then other thing is that we know that even Arabidopsis after 25 years, we have more than 50% genes for which exact molecular function is not known. For rice, many crops, less than 10% genes are experimentally characterized. So this also gives an opportunity to capture all that knowledge that is available for different crops. So for example, here is an example of a study from barley. And in barley, this gene EGT1 was found to be responsible for plant gravitropism, and the gene is not characterized in Oryza sativa or even Arabidopsis. And so we looked into their ortholog, and there are single ortholog across both species. And so we feel that this orthologous evidence is very good to use, and then we can predict the function. We can improve the annotation of plant genes. Also, we can see that difference in different plant species for pathways. So for example, here is the example of short day and long day flowering regulation in different plants. So we have ability to depict the pathways, different versions of pathways, and capture that diversity. More recently, we are using agentic AI for text mining and doing some of the gene summaries and pathway summaries, but we like to do it very carefully and a human in loop approach, and verify that we are not picking something that is not good. So we like to maintain our high quality data. And then we have currently 8,000 unique users that come to Plant Reactome portal. Some come through Grameen, and other comes from the other collaborating sites. Finally, I like to acknowledge tremendous help over the years from our undergraduate students who helped to curate, who were behind a lot of the curation, reading papers and discussion, and a lot of inspiration to me. And then again, I like to acknowledge Plant Reactome team, Pankaj Jaiswal as a co-lead on the Plant Reactome, and our fantastic software developer, Justin Elser, Parul Gupta, another curator, and our collaborators, Grameen, Reactome, EBI, and then some folks in Texas A&M and Cornell as well. And we like to acknowledge funding from NSF, NASA, DARPA, wherever we can get. So, and we like to thank also all other database users and data providers and all collaborators in ISB as well as in the egg biodata. Thank you very much.
Thank you very much. You are going to be able to ask Shushma and, of course, Aiton and the next speakers questions at the panel discussion. So thank you, Shushma. And now we are welcoming Dr. Maya Rai from Heidelberg Institute for Theoretical Studies in Germany. And she will be talking us about Sabio RK at 20 years, a modernized resource for curated biochemical reaction kinetics.
So thank you. Thank you very much for the nice introduction and sorry for the opportunity to give this presentation here. So Sabio RK is a manually curated relational database which was released for the first time in 2006 and contains biochemical reactions and their kinetics, and was originally developed to support systems biology, especially by offering kinetics data to create kinetic models of large biochemical networks and also to support the enzymology research as such. So 2 years ago in 2024, we decided to rewrite the Sabio RK code from scratch to get a modernized platform which is based on current open source technology. And our beta version was finally released in April 2026, so just currently. And in the next minutes, I will talk about our re-implementation and new features of the new Sabio RK. So first, what is reaction kinetics? Reaction kinetics is the study about the speed or the rates of biochemical reactions. And these are typically catalyzed by enzymes. Enzymes are binding to the substrate and thus lowering the activation energy and increasing the speed of the reaction. And before the substrates are released, or the substrates are converted to the products which are released, and the enzyme itself stays or remains unchanged. We are collecting not only the kinetic parameters itself, but also the information about the kinetic law type and kinetic law formula. So, but we are collecting all these kinetics data always in context. So it's this is our data model of a typical data entry or data record. In the center is always the biochemical reaction itself, which is composed of the compounds, so the reactants, the substrate products, and the modifiers like cofactors, inhibitors, or activators. If it's a natural occurring reaction, then it's part of a biochemical pathway, which is mentioned in our data entry. And for sure, it contains information of the catalyzing enzyme and its biological source, as well as the kinetic information itself and the experimental conditions under which these data were measured. Yeah, and the reference data is also included for sure. And typically, most of our data is derived from scientific publications. Currently, our database contains about 75,000 database entries being derived from about 8,000 publications. So all of our data is highly interlinked or linked to other databases and annotated with ontologies. And also many of these databases link back to Sabio RK as well, as for example, you find a link to Sabio RK under the link out in PubMed and in UniProt in the functions section under enzyme and pathway databases, and in Brenda under external links. So why did we decide after 20 years to re-implement Sabio RK from scratch? First of all, the code got more and more complex over the years, as every developer might know. but the major reason was that our database was somehow a fragmented one. So our legacy workflow here shows that, coming from the literature. So most of our data is extracted from literature. So we have master students, student workers who are extracting the data out of the literature and then inserted into our input data form, which is connected to a MySQL database. In the next step, we as a curator, so my colleague Ulrike Wittig and myself, we are rereading the same publication and doing and validate and augment the data with annotating and interlinking and adding new entities. And then the data is added to the input database. And then we had another complex transfer procedure where the new entities had to be submitted again. And then it was finally visible on the public interface. So because of, to get rid of this complex data transfer, which also led to some discrepancies between the input and the output database, and which had also no proper version control in place, we decided to rewrite Biocuration. And after more than 1.5 years of re-implementation, we did that together with an external company. We got finally a unified platform, which is based on open source stack. It is based on a JSON data model now, which we didn't have before. And it unifies the data input and curation and the public access in one unified system. And the technology is in the back end. We have only one PostgreSQL database now, and we utilize .NET 10 and SolidJS for performance reasons, and Apache Sol-R is used for search, and everything is containerized into Docker for reproducible deployment. And we also added the RESTful API for programmatic access, as is due to rate limitation in IP-based partitioning, quite secure for and prevents denial of service attacks. So now coming to the data entry side first, to our new features. We have here this guided curation workflow implemented in the dashboard, which helps us as data entry persons to guide us through the entire workflow. So first, the data is, or the reference is imported from PubMed, and the task is then assigned to the student workers who are doing the actual data input before the curator is working on the publication again. And if all entries of one publication are correct and together, then we can publish the data directly to the user for the user interface. And another new feature is that we can now directly during data entry add our new data entities here, for example. Sorry, it's not visible. But here we can see that when we enter a new compound, then we are directly guided to the details page where we can add the external links and INCHI keys, mile strings, things like that. And all changes are now tracked and can be easily seen in this changelog viewer. We have now also implemented the possibility to directly amend published entries. So when we get any feedback from the users that there was a mistake, we can go to our data entry site, make there the corrections, and republish the entry directly. Before, it always took a while until the entry was published. Now coming to the public interface. So here we have a new functionality, which is our browse function, where the user coming first time to our database can get an overview. What kind of data do we have? All the data are grouped, are categorized in these color-coded topic groups. So for yellow for all around organism, tissue, cell location, red is for the enzyme information, and blue for the reaction and the pathway information. And this color coding is maintained all over the data entry and the data application side. And by clicking here on the number, which just tells you how many entries we have, for example, this organism Homo sapiens, by clicking on the number, you can directly start a search. And this will then directly be included in the search field. And we have now, as a new feature, also implemented that the last 50 queries are saved. So you can always go back and start the same query again. Or you can also even define your own specially named query for the long term. And you can export and import these search queries as well. The default view of the result set is now also new, the Result Explorer, which gives, again, an overview and also a ranking. Here you see how many entries we have. Sorry, you can't read that again. This is how many that we have here for the search D-glucose 6-phosphate. We have so many entries for Homo sapiens and then ratus and species. So we are collecting data from all organisms. And you can here again refine the search directly by pressing on these numbers. And when you have entered all your search terms, you can also change to the entry view where all the entries are listed. And here you have you have also the option to select which entries you want to export in the JSON format, in XML format, or SBML format in this case, or as a table format. And by clicking on the number here, you get to the single data record, which looks now like this. So we have, again, the color coding. On the right side, we have all the links to other databases. And here we can also see the JSON representation of the data. And in comparison, this was our old single data entry view. So yeah, it's a little bit modernized and clearer structured now. Yeah, with this I come to the end of my talk and just want to summarize shortly that we have now this unified, modernized open source platform with about 75,000 database entries currently, and it was released in the April. And here you can scan the QR code and feel free to just try out our new database and give us feedback. We would be very happy. We have a contact form for that. And we are already working on the next release where we want to integrate our visual search, which was done in a former project called SabioVis. And we also want to allow customizable data views for users in future. With this, I want to thank our collaborators in the project, our student workers, and my direct colleagues, and you for your attention. Thank you.
Of Information and Systems, Japan. And he's going to be telling us about the development of the Togo MCP server to facilitate access to the RDF portal.
Okay. Okay, thank you, Chairperson. I'm Yasunori Yamamoto from Japan, so from far east to the far south, and I'm pleased to be here. It's the first time for me to come South Africa. I'm enjoyed it very much. Okay, so let's start for this title. Yes, so first of all, I briefly introduced the DBCS provided RDF portal. So the portal is developed by DBCS and NBDC, and it's a Japanese organization, and it has been developed and maintained for more than 10 years. And it has now 57 RDF datasets and also 198.5 billion triples. And so it's covered more from genes, proteins, and also compounds, and you name it. And so you can access to this data set by using a Sparkle query. So Sparkle is very flexible and powerful. So you can access to this data set and get exactly what you want by using a Sparkle query. But the Sparkle query is very difficult. So how many of you already use the RDF portal and the Sparkle query? So do you know Sparkle query? Oh, okay, yes. So do you think it's easy to learn Sparkle? Yes, yeah. And also I like the Sparkle, and almost every day I write Sparkle, but I know it's very difficult to learn, and the learning curve is very steep. So, so this is very difficult for the domain scientist. So the problem is, first, it's learning curve is steep, and also when you try to retrieve the data from RDF dataset, you have to know the exact identifiers or URI to get the appropriate result. And also, it is often the case that in biological case, and biological concept has multiple URIs, so it is very confusing to get appropriate data. Okay, so, okay. Yeah. To address these issues, we developed Togo MCP. So Togo MCP is an MCP server that supports AI agent in accessing the RDF data. And also, I'd like to emphasize on the 2 points concerning RDF Togo MCP. So Togo MCP doesn't process any natural language queries, and also the portal or Togo MCP does not generate a Sparkle query. Instead, the Togo MCP provides a structured context and services that the agent uses to perform these tasks. Okay, and so here's the 3 components that Togo MCP provides. So first, the Togo MCP has a rich metadata called MIE metadata. So I briefly explain data for MIE metadata. And so by using this metadata, an agent can produce or generate an appropriate Sparkle query. And next, ID resolution via APIs. So Togo MCP resolves user-relevant entities into correct identifiers. So user doesn't necessarily need to know the exact URI beforehand. And so the third component of the Togo MCP is that MCP host integration. So MCP-based integration, it can be realized by Togo MCP for the database accesses for domain scientists. Okay, yes. Yes, so I briefly introduce MIE metadata. So MIE metadata is a core of the Togo MCP, so it's very important stuff. And the Togo MCP is written in the YAML metadata, and this consists of these 4 parts. So sex schema and void metadata, and Sparkle examples, and also natural language annotations. So some of you already know that sex schema is briefly describes the data structure of IDF dataset, and void metadata provides the statistical information for the dataset. So this kind of stuff already curated and generated for each dataset. So the agent can use these metadata to generate the appropriate Sparkle query. So this means that the quality of MIE metadata is directly determines the quality of query generations. It's very important stuff for the Togo MCP. Okay, so there's a system overview here. So first, user makes some natural language questions. Then it submits to the agent such as Cloud Desktop. And the Cloud Desktop parse that natural language questions and extract the key terms from that question, such as gene name or disease name. And then the Cloud Desktop or agent submits that each key term into the Togo MCP, and the Togo MCP asks for the external API lookup services such as Togo ID or ontology lookup services provided by EBI. Okay, then after getting the appropriate identifiers. It fed into the agent by Togo MCP, and the Togo MCP tried to generate a Sparkle query based on the mere data for each dataset. And then it again proceed the Sparkle query into the Sparkle endpoint provided by RDF portal and getting the result from RDF dataset. Then summarize the result and back to the user by using natural language. It's kind of an overview of the MCP server and agent stuff. Okay. Yes. So let me show you some example here. So if the user submits a query like what proteins interact with insulin and are associated with type 2 diabetes here. So first, the agent parse these natural language questions and extract, for example, here it's kind of insulin or type 2 diabetes. And then the agent submits or asks Togo MCP, and Togo MCP looks up that kind of key terms and getting the corresponding identifiers and back to the agent. And also agent getting the mere data for each dataset like Uniprot or Mondo or such kind of databases and generate a Sparkle query. Then submits a Sparkle query into the Togo MCP, and the Togo MCP forward that query into the Sparkle endpoint of the portal and back to the result and fed into the agent of the result of the RDF data. So then the agent summarizes that data and back to the user to read the data in result in natural language. Okay, so Togo MCP acts as a catalyst to facilitate this process. Yes, okay. And so the Togo MCP functions as an enabling layer, and also the agent performs core reasoning and query generation. So if we want more information from this example, you can access to this URL here, or you can access by this QR code. So Cloud Desktop generates this result, so you can trace what happened from this process. Yes, okay. So the key benefits and contributions here. So the most important part of Togo MCP is lowering the barriers to the domain scientists to access to the RDF dataset. They don't need to use any Sparkle query, just issue the query in natural language. And also, agent can handle that natural language processing. So Togo MCP itself is language agnostic. So you can use any language you want if the agent can handle that language. So not only in English, but you can use any English or Japanese or Swahili or something. So if the agent can handle that, you can use. Okay. And also you can access to this here. So if you want more about the Togo MCP, you can learn more about this service. Okay. And also here, is, yeah, and summary and future works. So its time is very consuming and strict, so I only show you the future work. So we expand the dataset coverage beyond 24. Now, currently only 24 datasets are supported by Togo MCP, and we'll continue to expand the dataset. And also, if we want more dataset, please ask me. So you don't want, you want in this Togo MCP service, because once you get the mere metadata, you can ask for the Togo MCP by using a natural language. So any database can be welcome to our service. Okay, and yeah, so this is okay. Okay, thank you for very much for your attention. Yeah.
Thank you very much. Now we are welcoming the speakers to upstairs at stage for the panel discussion. Oh, sorry. Any questions for one of our speakers? Or for all of them?
Hello. My question's for Dr. Yasunori Yamamoto. Lovely presentation. I've been looking a lot into MCPs. My question is kind of two parts. First part is have you tested this with any other LLMs, specifically more underpowered LLMs compared to Claude, the commercial provider? And the second part is with your MCP server, you have the MIE and you have 24 data sets. It could be quite taxing on the context window. Have you found any limitations with the queries when you start to build out larger sets of queries that require more data sets from multiple APIs?
Okay, yeah. For the first question is we also tried not only for Cloud Desktop, but also ChatGPT and also the open source version of the Qwen 20.5, such as, yeah, and also we verified that works, but not well comparing to the Cloud Desktop. Currently, the Cloud Desktop is best. Yes, and for the second question, for the limitation, yeah, even though we developed metadata and we write many things into the metadata for feeding the context into the agent, but some agents still generate inappropriate Sparkle queries and also not well defined, totally meaningless queries, such a kind of stuff. So it's very difficult to, but gradually the performance is better, and also that model is also developed very fast. So we rely on that kind of development for the LLM. Yes, thank you.
Yes, thank you. I have a question for Dr. Maja. Majare, it's right for no. I just want to know, you have presented the workflow for data entry and data curation. In which step for the work in the workflow you ensure the quality of the data before publication?
Yes, what was exactly the question behind this workflow?
In which step in the workflow you ensure the data quality, I mean the accuracy of the data, the data is completed before publication?
This is what we do with our daily work. So first, we as curators go over the entire data, what was inserted by or input via from the students, and we check every data entry again. And yeah, when it's correct, then we submit it finally for publication.
Thank you.
Welcome.
Hello, my question is for Dr. Yamamoto. I'm just curious to know, did you try, apart from MCP, did you try any other technique like retrieval augmented generation to achieve what you already did? I'm just curious to know what the results look like.
Okay, yes, thank you for your question. Yes, we also tried RAG, yeah, but yeah, in my view, the RAG is not enough for getting the exact answer from RDF dataset, because the RDF dataset is very follow the ontology stuff, and there are many class and properties, and those stuff can be fed into the LLM by using a mere metadata like sex or void metadata. So if you want more appropriate and good result and question what you want, I think MCP stuff is better. Yeah. Thank you.
Any other questions? I can ask a question to Shushma. I have a question for you. I was wondering, I mean, some species, plant species are better covered than others. And I was wondering if you try to prioritize those that maybe there's no as much of information to look for them and to curate that, or is it you try to collect those that are more defined, so to relate to them via orthologs or... Sorry, you should... Yeah, exactly.
Yes, we have a couple of criteria. So the first criteria, we looked into literature. How much literature is available for which species? Then we looked into transcriptomic and genomics data. Then we looked into the evolutionary span of the species. So we wanted to include at least one representative from each family. And then we wanted to have crops, major crops and minor crops. And then we also wanted to have like the model plants for each each kind of lower plants, and maybe one for bryophyta, maybe one for tree species, maybe one for gymnosperm. So idea is to cover the whole evolutionary spectrum and have things, and at least one representative from each. And then once we curate this data from at least 15 species, it can be projected based on gene orthology to another 150 or 1,000 and whatever capacity we have.
I see. Thank you.
Thank you.
Yes, follow up for Dr. Yamamoto. Yeah, I'm back. So I'm very curious about this system that you built in the context of data sovereignty and privacy. I was wondering, how did you cater for that in your system? Say someone is not comfortable maybe sharing their metadata or something. Is there a way that your system can accommodate local infrastructure or large language models downloaded locally?
So far in plant sciences, we don't have much of the data sovereignty problem. As of now, it is, I think, more into the human and much of the ethical regions in the human. We believe in open data, and we think that we cannot work in isolation. And we try to I personally believe very much into the international spirit of the science, and we know none of us can actually do it alone. There is so much of data and such a little funding available. Database sustainability itself is a big issue, and then training. And so I very much believe that science is an international enterprise, and into the collaborating as much as possible. So, and we provide training for free. We advocate for the open access. We advocate for the collaboration as much as possible.
All right. Thank you. I wanted to hear from Dr. Yamamoto following the same thought. Your system, the one that you built, does it support locally downloaded large language models?
What do you mean by that? So you want to use your data in your PC?
Yes, yes. I want the model to be locally downloaded onto my PC so that instead of maybe Claude, can I substitute Claude for something that?
Yeah, I think so. If you put the configuration file for the Code desktop, you can use both the remote MCP server and local MCP server. So that your answer to your question?
Yeah,
and
in part.
Also, okay. It's okay?
Yes, yes, it's okay. Thank you.
And in addition to that, concerning your question before, I wanted emphasis on the RAG. So RAG can be used for getting exact identifier from the keywords. There are many databases like written in natural language. So if you try to make a query in the natural language, and the RAG can get any entry of the data set, so you can get the exact appropriate identifier by using a RAG. So I think it's kind of a very useful case using RAG.
I just also wanted to add that we don't capture any random data. We only use the published data. And also we believe that it is peer reviewed, gone through certain standards. And so this is how we consume data, whether it is omics data It has to be published part of any manuscript, and that is available as an open access.
All right, thank you so much.
Are there any other questions? Then I'd like to ask you something, Eitan. It's just a curiosity, really. I was just wondering, how does AGVD manage? Because I noticed that you gather information also from different resources and sources. So if there are changes, they update information from them, data. How do you? Is it automatically related to what happened in HB, or you have to?
No, it's manual. Actually, all of them. So the big job is the joint call set, and that we try to do every 6 months, because that's usually because there's more samples and more. And then we do the annotation as a job also manually. So
it's manual checking all over again. No. Okay. Okay, I think if there are no other questions, we can just clap our speakers for the last session of today. Thank you so much for sharing your work with us. And thank you everybody for listening very attentively. Housekeeping note, we are going to be having the shuttle bus at 6:30 at the lobby to go